So how many people will attend this meeting?
There are 120 candidates who have registered for this meeting, but some are on YouTube Live, and currently we are 57 in this meeting.
Okay.
Yes,
So
yesterday evening, it was very late. Until 7 o'clock, we are having the presentations, oral presentations of the participants.
Okay.
So I think they are a little bit delaying to join this in the morning session.
Mm-hmm.
Yes,
Nice.
live streaming is there. I think again they may visit that YouTube link. They may gather that information, whatever you shared.
Okay, nice.
Yes.
So will it being open to the public or just open to the people who register for this meeting?
Yes.
YouTube Live is open to the public, whereas
okay.
entry is registered to the participants.
Okay, I see.
We can start, Ma.
Yes. Good morning. A warm welcome to you all for the second day international conference first session. So dear delegates, all of you utilize the opportunity. Today we have Kulaju from Emirates. Mainly he's doing her postdoctoral research in food science and the food science department in the University of Manchester, Emirates. She received her PhD from the University of Manchester, Emirates, master's degree from Xi'an University, and bachelor's degree from Nanchang University in China. She almost published more than 29 research articles and in peer-reviewed journals with the citation of almost 575 citation. It's a good thing, ma'am. She has also received lots of honors and rewards from international and food institutions, including American Oil Chemists Society and Institute of Food Technologists. And she joined the group of Dr. David Julian Michael Clement's group in 2017 and currently working on characterization of plant-based foods and protein, including improving their appearance, texture, and nutrition related one. Today, she is delivering her talk on fortification of plant-based milk with calcium by reduce vitamin D bioaccessibility and in vitro digestion study. So I welcome you, Dr. Ju.
Let me share my screen. Thank you so much for the great introduction. Is the screen looks okay?
Yes.
Yes.
Okay, thank you. Again, thank you so much for inviting me to do this presentation. It's really my honor to be here. My name is Hualu Zhou. My PI is Dr. David Julian Mike Clements. Here, I want to introduce you my recent study, fortification of plant-based milk with calcium may reduce vitamin D bioaccessibility, an in vitro digestion study. First, I want to introduce you a little bit about the University of Massachusetts Amherst, which located in the northeast of United States. The food science department has high reputation in global and rank #1 in the United States. Plant-based milk has developed to replace the cow's milk. It is very challenging to do that because cow's milk has high quality and people used to its appearance, nutrition, and facility. So why do we want plant-based milk to replace cow's milk? because we are trying to improve the sustainable to food supply and animal-based food products and not cause the environmental damage. So plant-based milk become more popular, and there are many kinds of plant-based milks, such as soy, pea, oat, coconut, almond, and rice. Despite their well-established environmental and ethical benefits, there has been some concerns about their nutritional profiles. For example, many plant-based milks are deficient in important micronutrients like the vitamin D and calcium, which could result in long-term health problems if they are not obtained in sufficient levels from other sources. Before we talk more about the nutrition, let's see how do we make plant-based milks. The conventional method of making plant-based milk is by size reduction, which usually starts with some plant-based materials which have oil sources in it, like the soybeans. What we often to do is use this one is taking it, sucking and grating it and breaking it down to the plant tissues and the structures and separate out the soluble components. We may hydrolyze it and homogenize it to break down the structure more. We may use heat treatment to kill the enzymes and bacteria, and we may add some other ingredients like the stabilizer into the system. At the end of it, it will have very complicated colloidal systems. We have the oil bodies, we have some tissue fragments and the biopolymers in it that give the texture and appearance of the product. Other way we can make plant-based milk is by homogenization method. We start with the oil and the emulsifiers, and we pass through the homogenizer to make the we first mix them together to form the coarse emulsions, and then we pass through the homogenizer fine emulsion, and again do some like thermal processing and add some ingredients to keep them stable, and then do their packaging. Again, this is also the colloidal system, and the fat droplets were covered by some plant-based emulsifiers. There are also lots of plant-based ingredients we can use to formulate those products. We have the plant-based oils and plant-based emulsifiers. For the emulsifiers, there are some surface active molecules can stabilize the droplets in the system, like the protein, polysaccharides, and biosurfactants. So we can get them from the soy, sunflower, corn, pea, and the same for the plant-based oils. The research we have been working recently is using emotion-based delivery system as a vehicle to deliver sort of nutraceuticals, minerals, and vitamins in the food products. To claim health benefits, plant-based milk or emotion-based delivery system is an ideal vehicle for doing this. We can incorporate different bioactives like the hydrophobic ones and the hydrophilic ones into the oil together. So we can make an emotion-based delivery system and added it to fortify the plant-based milk. There are also different types of bioactives we may want to put into the plant-based milk. So we can put the nutraceuticals like the curcumin, carotenoids, and other nutraceuticals claimed to have health benefits. We can coat the omega-3 fatty acid, the protein also in the liver system, the probiotics and the vitamins and the minerals are maybe we don't have enough of them in the vegetarian diet, but it is often challenges to do this. Some of them are not soluble in water. Some of them are easy to crystallization, like the curcumin and the carotenoids. Some of them are unstable, like the vitamin D. They may degrade during the storage, and some of them get very low bioavailability. So for the nutritional fortification, on one hand, we want to make sure the emulsion delivery system we add to the plant-based milk still maintain good physical chemical properties and stability of the final product. On the other hand, we should pay attention to their gastrointestinal fate and try to increase the number of micronutrients can reach into the small intestine and have high bioavailability to increase the efficiency of the fortification. Here, I want to briefly introduce the gastrointestinal environment. Typically, we can divide the human gastrointestinal tract into 4 regions, including the mouth phase, stomach phase, small intestine, and the colon. In the mouth phase, we have like amylase to digest the starch, and some food matrix can be broken down when we chew it. In the stomach phase, the gastrointestinal fluids are acidic. The pH could be around from the 1 to 3. We have gastric digestive enzymes like the pepsin and the gastric lipase. They can digest some protein and lipids. In the small intestine phase, most of the macronutrients can be digested, and the nutraceuticals can be released and absorbed in this phase. In the colon phase, many bacteria can found, and we hope we can deliver some beneficial nutrients to the bacteria and bring health benefits to humans. Therefore, for our study, we used a standardized in vitro digestion model developed by the InfoDress community. We take our sample and put them through a simulated mouth, stomach, and the small intestinal phase. We use a pH state to measure the lipid digestion or protein digestion at the end of the process. We got at the end of the process, we will centrifuge it. And if the fat is not fully digested, we got a lipid top. We may also have some bioagents solubilized here. We have the micelle phase, which we think is the mixed micelle. And this is where we assume the bioactive agents is going to be informed and accessible for the absorption. And then we will have some insoluble matter. This could be the calcium salts of free fatty acids or bile acids. And this may also contain some bioactive agents. So we assume that the bioactive agents in the lipid phase and the sediment phase is not bioaccessible, but the amount in the micelle phase is bioaccessible. In this study, 2 micronutrients, vitamin D and calcium, were chosen to fortify a commercial almond milk. From the nutrition label, we can see it has low and almost 0 gram of vitamin D and calcium in the initial product, which may induce vitamin D deficiency if they are not obtained in sufficient levels from other sources. For the vitamin D, we can use the narrow emulsions to encapsulate the vitamins in the oil phase. And for the calcium, we can directly encapsulate it on the aqueous phase. However, for the calcium, more attention should be paid because calcium can play an important role in digestion process. It can act as cofactor of enzymes and accelerate the lipid digestion in small intestine. But the cationic calcium ions may also induce the particle flocculation and affect the plant-based milk quality and reduce the bioaccessibility of vitamin D. Here is our experiment design. First, we use the plant-based oil, the corn oil, and the plant-based emulsifier to fabricate the vitamin D narrow emulsion. And then the impact of different calcium type, the soluble calcium and insoluble calcium, and also the concentration of the calcium on the plant-based milk, physical chemical properties, the lipid digestibility, and the micronutrients bioaccessibility was examined. So first, the impact of adding vitamin D narrow emulsions to almond milk was investigated. Different amount of vitamin D enriched narrow emulsion, like the 0%, 10%, 30% to 100%, were well mixed with almond milk. Here, the 0% group is the pure almond milk, and the 100% group is the pure vitamin D narrow emulsions. From the picture and the total changes in the color values, we can see that the incorporation of the vitamin D enriched narrow emulsion only had a minor effect on the overall appearance of the plant-based milks. Next, more initial properties of fortified milk samples were measured, including the mean particle diameter, the surface potential, the pH values, and the macrostructure. After adding the vitamin D narrow emulsion into the almond milk, which are those 10%, 30%, and 60% group, the system particle diameter were increased. Also, from the microstructure, we can see that the particle in the mixed systems were highly aggregated. We hypothesized that the particle aggregation may have occurred due to a change in the pH when the plant-based milk and the narrow emulsions were mixed. The narrow emulsion had a much lower pH than the commercial almond milk. So there was a decrease in the pH of the mixed system as more narrow emulsion was added. From the viscosity data, interestingly, introducing 10% or 30% of the fortified narrow emulsion did not have a significantly impact the viscosity of the plant-based milk, but adding 60% lead to a large decrease in their viscosity. We also tested the 7-day storage stability by measuring their appearance and the particle size change, and notice the almond milk samples has the tendency for aggregation, which is consistent with the advice of many plant-based milk products to shake the product before opening the container. So in summary, considering all of those initial properties change after adding the vitamin D narrow emulsions, we found at a small amount, like 10% of the vitamin D narrow emulsion, will not significantly affect the quality of almond milk. Next, calcium was added to the aqueous phase of almond milk, and we passed them through the InfoGest in vitro digestion model to determine their gastrointestinal fate, as well as to determine the bioaccessibility of calcium and vitamin D. All samples was at 10% of the vitamin D narrow emulsions, and the last 4 samples were added 1 gram or 2 gram, like calcium chloride or calcium carbonate per serving, which is following the instructions by the FDA.
Okay.
Here, the sample 1, which is the narrow emulsion stabilized by surfactant 20, was used as a control. All the 6 samples has the same oil concentration and vitamin D concentration. First, we measured the size and surface potential of each sample before and after digestion. We observed that the initial mean particle diameter of the controlled group was around like 100 nanometer and remained relatively small throughout the whole gastrointestinal phase. The almond milk sample has a relatively large mean particle diameter, and the diameter increase after adding the soluble like calcium chloride. That probably because of the cationic calcium ions are electrostatically attracted to the surface of the anionic plant tissues and oil droplets, which is consistent with the observed reduction in the negative surface potential in the presence of the calcium. For more clear information, we can see from the confocal microscopy images. Here, the red color means the oil droplets of the sample. From the images, we can see the control group contained relatively small lipid-rich particles throughout the simulated gastrointestinal tract. However, all the almond milks contained relatively large particles within the initial samples. After the mouth phase digestion, the mean particle diameter of all almond milk samples followed a similar trend as the original samples. The sample contained the soluble calcium, like the calcium chloride one, has much bigger like particle size. After the stomach phase digestion, all the almond milks has the similar mean particle size diameter, which was attributed to the acid conditions, which is pH 3 in the stomach. After the small intestine digestion, the sample with no or like the low calcium levels contained relatively small particles, but those who added a high amount of calcium contained numerous large particles. We can see from here, those very large like lipid-rich aggregates are likely to be the insoluble calcium soaps, liposomes, or the undigested oils. The impact of the calcium form and the concentration on lipid digestion in the almond milk samples were established using the pH state method. We clearly observed that the addition of the calcium can accelerate the lipid digestion rate at pH 7. And the lipid in all samples was fully digested from the free fatty acid release amount at the pH 9. After the digestion in small intestine phase, we will centrifuge the solution to have a clear micelle phase. And the vitamin D concentration in this micelle phase was treated as the bioaccessibility. From the left figure, we found that the vitamin D bioaccessibility was higher in the control group, which is the fortified 220 narrow emulsion, than in the rest of the almond milk samples. And that is decreased when we like increasing the calcium levels in the almond milk. So what is the mechanism behind that? The right photo is the appearance of the digester after the centrifugation. We can observe more sedimentation with the calcium concentration increased. This means the addition of the calcium can help to form the calcium soaps. So the inhibiting mechanism we proposed is that the added calcium in the almond milk will release calcium ions during the digestion, and the cationic calcium ions will interact with the anionic like mixed micelles and generating insoluble calcium soaps. Therefore, some of the vitamin D will trapped inside the precipitates from the sedimentation amount, and the confocal microscopy images can both prove that. Another possible reason is that it might not be possible to remove all of the vitamin D from the digester phase using a solvent extraction method. The calcium concentration in mixed micelle phase was determined using the X-ray. The calcium bioaccessibility increased as the amount of the calcium increased. Interestingly, the insoluble form of calcium like carbonate, lead to a higher bioaccessibility than the soluble form, the calcium chloride, when added at the same level. This has been due to the differences in the ability of the different form of calcium to interact with the mixed micelles in the small intestine. From the bioaccessibility value from here, and we also noticed a large amount of the calcium that had been lost during the pass through the simulated gastrointestinal tract, result in the formation of the insoluble calcium salt. In conclusion, we use the vitamin D and the calcium to fortify the almond milk and explored the calcium effects on their gastrointestinal fate and the bioaccessibility. We found that the calcium chloride promoted the aggregation of the particles in the almond milk. The high levels of calcium reduced the bioaccessibility of the vitamin D. The bioaccessibility of the calcium increased with the increasing of its concentration. Those findings are useful for the development of nutritionally fortified plant-based milk with improved physical, chemical, and nutrition profiles. At the end, I want to thank my PI, Dr. David Julia Mike Clemens, and my lab members who give me strong support during this project. This is my email address. And besides the plant-based milk fortification, my research interest also including the food emulsions and the nanotechnologies to like encapsulate the nutraceuticals. And we also focus on the food digestion and the gastrointestinal tract. Besides the plant-based milk, we also pay a lot of attention to the plant-based meat, plant-based egg, and to characterizing the plant-based proteins. So if you want to know more information about me or you want to like collaboration in the future, feel free to email me. So thank you for your attention and welcome any questions and discussion. Thank you.
Thank you, Dr. Zhou. I ask the participants to unmute yourself, and if you want to have ask any questions to Dr. Zhou, you can directly ask, or please type in the chat box so that we can read it out. Before we get questions from the participants, I have one question. I want to understand more about it. You mentioned that you have measured calcium bioaccessibility using X-ray.
Yeah.
X-ray spectroscopy.
Yeah, X-ray spectroscopy.
Can you elaborate a little bit more on that? Because we work in that area, so I thought that will be useful for us. This particular aspect, yeah.
Thank you. This is a good question. Actually, I'm not like a professional, like a specialist in the X-ray. We just use X-ray to measure the concentration of calcium. So the first thing we can do is we will have a standardized curve of the calcium concentration in the digester. After we got it, we can use it to measure our sample. It's very easy to measure. You just prepare the solution and you put it in the X-ray, and the instrument will give us the result. Yeah.
At what stage you have used this for measuring the calcium content?
I think you just you don't need to like like pre-treat the sample. You just directly use the solution, which we use is the digester, and they can help us measure the calcium amount from it.
Okay. So from the samples of the digester, you have measured the calcium content using X-ray.
Yeah, that's true. We first prepare a standard curve, and then we can.
Okay. Okay. Thank you. Thank you so much.
Thank you.
Any other participants? You have any questions? Please mute yourself. All the participants are requested. Only if you have questions, please unmute. Otherwise, mute yourself. There's lots of background disturbance.
Joe, I have a question. So which one you have taken as a control in order to compare this almond milk bioavailability, bioaccessibility?
You mean the vitamin D?
Control, yes. You have mentioned that there is a twin control.
Yeah, that's true. Thank you. Yeah, for the control group, we use the twin stabilized narrow emulsion. So the emulsifier is the twin 20, which is a surfactant, and the oil is the same, is the corn oil, and its particle size is around 100 nanometers. Because the emulsifier is twin 20, so it will not, affect their digestion phase or something. So it would be a very good control. Thank you.
What percentage of insoluble calcium you have observed in the gastrointestines? So that whatever you have mentioned. The percentage of insoluble calcium.
You mean the percentage of the calcium we added?
Yes. Yes.
Right. So here we have like 6 groups. For the rest of 4 of them is the group added the calcium. You can see here, for all of them, we add the same vitamin D concentration. And for the calcium amount, they are different. For the group 3, we add group 3 and 4, we added 1 gram of the calcium per serving size of the plant-based milk. For the rest of them, we add 2 grams of it. Like we studied 2 concentration of the calcium.
Okay.
We added this.
So how much percentage has been absorbed of calcium? That is, in which form it has been absorbed totally into the?
Yeah, we also measured that. We can see from this finger, these figures, we can see. Oh no, this is vitamin D. Sorry. Yeah, from this figure, we can see that they have different percentage of the the calcium bioaccessibility in different samples, and the highest one is around 40%.
Yes. That is calcium carbonate, almond plus PD plus calcium carbonate. Okay.
Yeah, the calcium carbonate.
Okay, thank you. Very nice presentation.
Thank you.
Any other wants to interact? Or if you have any questions, please drop in the chat box. If we don't have any questions, Sirisha, are you there, ma?
Are you trying only with the almond milk or any other milk, soya milk or any other milk? You are trying only with almond milk, plant-based milk. Are there any other milks that are available?
Yeah, actually, they has a lot of available plant-based milk in the supermarket or grocery, like the almond one, the soy one, or like the oat milk. For this study, we just use the almond, but in the future we can like include more kinds of plant-based milk.
Any other questions we have? Generally, from a pharmacist perspective, we avoid calcium in the formulations because of its ability to complex with it easily. Bioactive molecules, it complexes with calcium easily and prevents absorption of the bioactive molecules. But it is really interesting to note from your work that vitamin D and calcium can go hand in hand. And even they're showing bioaccessibility, that it is a new learning for me from today's your presentation.
Great. Yeah.
Okay. Sirisha, I think, shall we conclude?
Yes, yes.
Okay. So to conclude this session, I extend my heartfelt thanks to Dr. Hola Zhu, postdoctoral research, University of Massachusetts, Amherst. We are extremely sorry if we have any pronounced your name or your university in a wrong way. We're extremely sorry for that pronunciation. But your presentation was excellent. It was in a very scientific way, and I believe that our students might have learned how to do the food technology research in a more scientific manner, because you have brought in lots of spectral analysis data, lots of in vitro data, actually that in vitro models we generally use in pharmacological evaluations. So that was very interesting, and we really extend our heartfelt thanks for accepting our invitation and be part of this conference. Thank you, Dr. Holazu, and we look forward to work with you. Thank you.
You're welcome. Yeah, you're welcome.
Here we have Professor Aruna, head of the Department of Home Science, and we have Professor Pushpa Bharati. Madam has retired from Dharwad University. I think Dr. Holazu has just left, madam. Okay, thank you, Sirisha. I think you can move ahead with the next session.
Yes. Good morning, madam. Good morning to you all. Here, Pushpa Bharati madam is there. So we will start next session with the introduction of the madam. I welcome Niharika to introduce Pushpa Bharati ma'am. Welcome you, ma'am.
Thank you, madam. Madhavi akka, share the screen.
Yes, sharing. One second.
Good morning, madam. Good morning, all the delegates. It's my pleasure to introduce for me today's speaker, Dr. Pushpa Bharati, retired professor from University of Agricultural Sciences, Dharwad. Madam has 33 years of experience as a professor and a dean of College of Community Sciences, University of Agricultural Science, Dharwad. Madam's specialization was in food science and nutrition, and her area of research is micronutrients, especially minerals, food and nutrition security, community nutrition, underutilized fruits and vegetables, minor tuber crops. And madam has published 82 papers in national and international journals, and published 9 books. In that, 58 articles were very popular. Madam has completed 7 major and minor research projects as a principal investigator and co-principal investigator. She completed 7 projects. Madam has a member in various committees from reputed universities like a BOS member, University of Mysore, and BOS member, Akka Mahadevi Women's University, Vijaypura, member of academic body Group, Guntur, member of selection committee for CAS, Maharani Saraswati University, Ajmer, Rajasthan. Madam guided 5 PhD students and 30 M.Sc dissertation students. Madam received 3 awards. One is ICAR Jawaharlal Nehru Award for Best PG Research in 1999, Young Scientist Senior Award of NSI in 1997, Best Paper Presentation Awards in 2000 and 2004. Madam act as a reviewer of various journals like Karnataka Journal of Agricultural Science, Journal of Farm Science, Mysore Journal of Agricultural Science, African Journal of Food and Nutrition. Welcome you, madam, for today's session. Now the session is hand over to you, madam. Thank you.
Thank you so much for the nice words.
Thank you, madam.
Thank you. Now, can I share my slides?
Yes, madam. Yes, madam.
Can you just enable me to share the slides? It's showing
Okay, just
as
a minute.
visible.
Now you can share, ma'am.
Okay, fine. Can you see the slides?
Yes, madam.
You are able to see?
Yes, madam.
Okay. Can you hear me properly, like audibility?
Yes, ma'am.
Yes, madam. Yes,
Okay,
madam.
fine. Thank you so much to the Srimati Padmavati Women's University to Home Science Department for considering me, especially Sirisha, Dr. Aruna, Dr. Jyoti, and all others. So before I start, I just want to acknowledge the the consideration for my this thing. So before I begin, I welcome you all to the University of Agriculture Sciences. Though I'm retired, I'm very proud of my university. So that on the occasion of this international conference, virtual conference on nutraceuticals and functional foods for human health, I'm going to talk on millets in human health. So this topic I have chosen for a few reasons. One is the year 2023 is going to be the International Year of Millets, as has already been mentioned by Honorable Prime Minister of the country. It's going to be the International Year of Millet, so that is one reason. And secondly, we have been working on millets right from 1990. So though we had stopped working for a decade, then again, we took the strings, and from 2001 onwards, we've been continuously working on millets, and we have completed a lot of projects on millet. So that is another reason. So I have tried to compile the data which I have from the department on millets for human health. So when we talk about millets, they are the first crops, because history says that they have been there since long time, even before Christ, and they are also the future crops. So there is nothing like a day may come when we will stop consuming the wheat and other things, and we may go in for millets, because now we have seen that people are talking about millets and they are becoming so expensive these days. So if we look into the history, there is a evidence of cultivation of millets in the Korean peninsula that is around 3500 to 2000 BC. In India, it is especially Foxtail Millet, Barnyard Millet, and Black Finger Millet have been mentioned in the Yajurveda texts, which are very old. So this shows that how old they are there, and since ages they have been consumed. And millet consumption is very common. It is old as Indian Bronze Age, that is 4500 BC. So this shows that millets have been very commonly consumed and they are there in the history. But in between they got vanished and they are now appearing again. We are revisiting these millets. So why millets have to be consumed? Why do we call it, consider it as such an important grain? But the thing is, millets are called as, they have got the name because 1 million grains can be held in a fist. One fist, we can hold 1 million grains. They're so small. So they are rightly called as millets, and they are rich in benefits, both health benefits as well as nutritional benefits, and they are good for food as well as fodder. For the animals also, they turn out to be very good fodder crops, and they're cultivated in unfertile land. They do not require much inputs, and without much effort, they can be cultivated. So they are good for nutrition, and they compare very well with rice and wheat, the most common, most exploited grains. They are superior in protective nutrients, especially vitamins, minerals, dietary fiber, and so many nutraceuticals and pharmaceuticals or phytochemicals are present in millets. Now, due to the, just a second. Okay. Due to the high resistance against harsh conditions, these millets are sustainable to the environment, and they are environment friendly, farmer friendly, and they provide cheap and high nutrient options for all. Nearly 40% of the food produced in India is wasted every year, but this is not so with millets. Millet do not get destroyed easily, so they can be consumed for after 10 to 12 years also. So they do provide the food security, and they play an important role in keeping a check on food wastage. So the lot of food wastage can be avoided with millets, and it is fibrous and has lot of minerals like magnesium, iron, calcium, niacin, and they are gluten free, and they are comparable in the protein content. But the advantage here is the amino acid profile of millets is much better than the regular cereals. So considering all this, the millets are called as nutritious and therapeutic grains. So, so what are these millets? They are classified into, based on the size, they are classified into major millets and minor millets. Sorghum, pearl millet, and maize are considered as major millets, whereas finger millet, foxtail millet, leaky millet are the exploited minor millets, whereas barnyard millet, proso millet, kodo millet, and brown top millet are the underexploited minor millet. There are to this list we can also add teff and other things. They are not millets actually. They are the pseudo grains. They are not. They do not come under millet. These are the major millets, and these are the minor millets like foxtail millet, little millet. This is the kodo, barnyard, pearl millet, proso, and finger millet or ragi. So these are the vernacular names of the millets, like in different languages, Indian languages, what are they called? So this, I don't want to go through all these things because you get it easily. Let's see one by one a few millets. What is the importance of these millets? One is sorghum. Sorghum, which is fifth major cereal crop, and it is the stover of this crop is highly valuable as a fodder for the animals, and they are used for production of alcohol and many other industrial products. They are grown in Maharashtra and Karnataka basically, and also in North India. Pearl millet is grown in North India, and this is sixth major cereal. North India, it is very common. 90% of the acreage is in North India. And there are different types of pearl millet: pale yellow, brown, gray, slate blue, or purple. There are different colors which are available. Then finger millet is southern India, crop of southern India, East and Central Africa. And most commonly it is consumed in the form of malt, especially for the children as a weaning food. Then foxtail millet, which is also called as the Italian millet. The name goes by the earhead, which looks like a tail of the foxtail. So it is third largest crop among the millets, and it is cultivated in Europe, North America. This is commonly grown as a bird grain. It is used as a bird feed, and this contains the outer husk, and it has to be removed just like paddy. And then after removing the husk, we get the rice, that is foxtail millet rice, which is ready to be used or cooked. So then the little millet, as the name says, this is called little because it is very small. The grain is very small, maybe the size of semolina. It is common in eastern Ghats of India, and the stover is very good fodder for the cattle. And another advantage is little millet, when it is highly polished, it is white in color, but unpolished grains are available these days, and they are bland and suitable in many products. Then comes the kodo millet. This also has the husk, and it has to be removed. In India, it is domesticated around 3,000 years ago, and kodo millet is commonly grown in Uttar Pradesh, Kerala, Tamil Nadu, and parts of Karnataka. And then barnyard millet is another one which is called as Japanese barnyard millet also. It is also having good food and fodder crop, and this also has the husk, and it has to be removed to get the rice. Then comes the proso millet. Proso millet is a short season crop. It can be very easily cultivated, and after hulling, nutritious palatable cereal is used for unleavened bread, or it can be cooked like rice, and it can be used for many preparations also, and it is commonly used in northern part of India. Then brown top millet. Brown top millet is not very common. It is underutilized millet, and it is got the name because of the color of the earhead, brown top. So it is called as a brown top millet. It is native of India and commonly grown in Karnataka and Andhra Pradesh. So with these brief outlay about the millets, now let us see the nutritional benefits of these nutri-cereals. They are nutritionally superior to many finer cereals, especially in protein, minerals, vitamins, and fiber. In comparison to polished rice, the protein content and fiber content is very high in foxtail millet, especially it has got 8% fiber. Then good source of micronutrients, and energy is almost similar to the other millets. Fat is higher especially in pearl millet and foxtail millet. Protein is higher in foxtail, barnyard, and kodo millets compared to rice. And these the carbohydrate content is less in millets and very less in foxtail millet, and it is a better food for the management of lifestyle disorders. The thing is, carbohydrate, whatever is present in these grains, it is in the complex form. So complex carbohydrate, which is converted into, which is more, part of it is resistant starch. So it is more beneficial for lifestyle disorders, especially diabetes, cardiovascular disease, and obesity. On an average, resistant starch is 1.6 grams per 100 grams. Then nutritionally, the fiber content is more. Fiber content is 6 to 7 times higher in these grains. So this fiber helps in the reduction of cholesterol, blood sugar, which is very essential for the management of diabetes and other lifestyle disorders. Fiber also helps in the prevention of constipation and colon cancers. Millets have considerable amount of minerals and which is required for blood formation and cell structure maintenance. High polyphenols, which helps for antioxidant capacity. Then calcium is another mineral needed for bone development. Especially we know that finger millet is rich in calcium. Zinc is another mineral which we are talking about these days. It is needed for many bodily functions, especially for the action of the enzymes. Then health benefits of millet. They are also powerhouses of antioxidants, flavonoids, anthocyanins, saponins, and lignins. Because of these antioxidants, they play a role in controlling the cholesterol level. They lower the cholesterol, reduces the cancer risk, and are effective in preventing or managing lifestyle disorders. They contain high amount of fiber, essential amino acids, and fatty acids, B complex vitamins, and vitamin E. Regular consumption of millet helps for the postmenopausal women who are suffering from cardiovascular disease, especially high blood pressure, high cholesterol, and breast cancer. It provides protection against breast cancer. Millets are high in minerals, especially finger millet is highest. We all know that it has got 344 milligrams of calcium as against maybe 30, 35, 50, up to 50 in other cereals. Then children's intake of whole grains, especially millets, is not advantageous because it reduces the absorption of minerals. Then milling, this is the study conducted in our department on 91 varieties or land races of little millets. The milling reduces the alpha tocopherol, then bran, because bran contains higher alpha tocopherol and polyphenols compared to decorticated grain. So antioxidant activity of whole grain ranged from 19 to 24, which reduced to 13 to 23 after milling. So after milling and removing the husk. Bran contained highest antioxidant activity compared to whole grains. Then millets are also helpful for kids and babies. They are rich in antioxidants. They help for muscle development in children, and they are rich in fiber, so they prevent constipation. And ragi being rich in calcium, it is good for bone development. So it is used as food for in the form of malt. It can be used for the children's weaning food. Then it is rich in iron. The millets are rich in iron, so they prevent anemia in babies and kids. So other advantages of nutri-cereals are they are ecologically safe, they are sustainable, less cost of cultivation with very minimum cost of cultivation. They are adaptable to climate change. They can survive and yield even in harsh conditions where other crops cannot survive. These can survive. They are less, they are susceptible to less pests and diseases. So they are free from pesticide residues. So we can say that millets are organic foods because we don't spray much pesticides or insecticides. So they are good. We can say they are organic foods, and the products prepared with millets are highly acceptable, nutritious foods. Especially we can make any type of foods with the millet, maybe sweet, savory product, bakery, or any kind of foods can be prepared. Now let us see a few disorders where millets can be used. Millets in diabetes. because millets contain high fiber, polyphenols, and antioxidants. So they are efficient in improving the glycemic control, decreasing fasting and postprandial blood glucose concentration. They reduce the insulin index and insulin resistance, and they lessen the glycosylated hemoglobin. Different cooking methods like steaming and baking, and boiling increase the GI up to 18.4, 16.3, and 11.3 units. Despite this, overall GI of millets is 52.7 approximately. So this comes under low GI foods. Consuming millet-based diet for long periods significantly reduced the HbA1c, glycosylated hemoglobin, in both pre-diabetic and diabetic subjects. This is the study which has compiled, a review article which has compiled 19 studies has shown this. Then in cardiovascular disease, they reduce the total cholesterol, triacylglycerols, and the body mass index. So because of this, they'll have less effect on the cardiovascular disease. They are good for the heart health. Consuming millets reduce the total cholesterol by 8%, so that it reduces from high to normal level in the people who have been included in the study. There was nearly 10% decrease in low and very low density lipoprotein and the triacylglycerol levels. Through these reductions, the level went from above normal to normal range. In addition, consuming millets decrease the blood pressure, with diastolic pressure decreasing by 5%. So, and then constipation, we know, everybody knows that dietary fiber improves the digestive function, and the fiber will help in relieving the constipation, because fiber combats constipation, flatulence, bloating, cramping, regularizes the bowel function, improves the overall health of the vital organs like liver and kidney, and it boosts the immune system. Then in weight reduction, we know very well when the food is rich in fiber, the consumption is less. That's one thing. Consuming millets reduce the BMI by 7% who are overweight and obese people, and it brings down the BMI towards normal. This is because millets have high satiety value. Like this is our own experience. When we consume the millet for breakfast, we don't feel like having lunch also. So that is the satiety value of the millet. So when the food consumption decreases, naturally the body weight also decreases, but simultaneously the person doesn't feel tired or doesn't have low GI. I mean, no blood glucose, hypoglycemia. It doesn't go into hypoglycemia. So all results are based on consumption of millets for 50 to 200 gram of millets per day for 21 days to 4 months. There was a decrease in the BMI. Now, these are all the benefits of millets and how much they are beneficial to the human health. Now, you must be having a question as to how the millets have to be consumed. Whether I have told you in the beginning slides that some of the millets have husk and they have to be removed, like and converted into rice, just like paddy, and then it has to be consumed. So just to show some of the technologies which have been developed in the department, value-added technologies, these will show how the millets can be consumed in regular diet. So these products which have been developed, like traditional foods, health foods, novel foods, baked foods, instant mixes, and snacks, these will show that the millets can be made part of the meal for every group, every population, every aged person. So first is traditional foods. They can replace the rice and other cereals. Jowar, rice, and many other cereals can be replaced except wheat. Wheat, because wheat is having gluten, and millets are devoid of gluten. So they do not contain gluten. So they are suitable for people suffering from gluten intolerance or celiac disease. So millets can be used for the preparation of products like bread also. That is, sourdough bread can be prepared. Millets can be used with other cereals, or they can be totally replaced. Now, a few examples are which have been done in the department. This is the products of pearl millet. Like whole grains can be converted into semolina of different sizes or flour. This is the work of Dr. Kashibai of the department. Then the millets can be converted into pops. They can be popped, different varieties, and into different products. Like the pops can be spiced and converted into snacks, and pearl millet can be prepared into khichdi, or this is paddu, we call it. It's also called as, I don't know, in different names it is called in different parts of the country. But it is something like dosa batter, which is baked or with little oil roasted. That's called paddu. And these are the different products and their acceptability index. Highly accepted is the khichdi, followed by paddu and spicy pops. Then this is little millet paddu, which is a breakfast item, very highly remunerative also, and it ferments very easily, and it requires less of black gram dal. We don't need to add a lot of black gram dal. These are some of the traditional products of little millet, which can be prepared for the regular use in the day-to-day life, like upma, bisibede bhat, or thalipattu, paddu, then curd rice, khichdi, they can be prepared. Then foxtail millet-based traditional foods like dosa, kheer, poha, or rice flakes, foxtail millet flakes, and then paddu. Ragi-based foods or finger millet-based foods, paddu, then dosa, then thalipattu or roti, and idli can be prepared. Then health foods like diabetic foods, supplementary foods, sport food, and parboiled millet. Diabetic food, this is the product which is prepared under the NAIP project, that is National Innovative Project of the department. This product is sold in the market, and the GI of this product, foxtail millet-based diabetic mix and little millet-based diabetic mix, GI ranged from 47 to 56 for all these products. This is foxtail millet-based food. This is barnyard millet-based health food. And this is little millet flakes, ready-to-cook flakes, and khakra. Khakra is the traveler's treat. Travelers can take it on the journey. They can consume it with maybe a little pickle or something. So these products have shelf life of 3 to 6 months, and with a GI of 47 to 56, which is low GI. This is parboiled little millet, which is raw grain and the parboiled grain or hydrothermally treated grain. The grain looks brown here, but once it is cooked, it looks white. The cooked rice is white in color, and the rice will be, the grains will be separated. And then the salient features of these foods, foxtail millet diabetic mix provides same calorie, 379 calorie, 13% protein, and 23% fiber. So this has the GI of 50 to 54. Then little millet, that is hydrothermally treated little millet, has GI of 57. Parboiled little millet provides good amount of dietary fiber, that is 19 to 26, and it is having GI of 57. And the consumption for 30 days reduced the serum blood glucose, cholesterol, triglyceride, and glycosylated hemoglobin. Then barnyard millet health mix GI is 59. It can be stored for 3 months in aluminum laminated pouches. Then RTC flakes, ready to cook flakes, they have dietary fiber of 22, and the significant result here is it is rich in iron, having 32.23 milligrams of iron per 100 gram with a GI of 52. So The effectiveness of this technology says that inclusion of these products in the regular diet for 30 to 45 days exhibited nutraceutical benefits, like it improves the glucose carbohydrate tolerance, reduces the plasma glucose, fasting glucose, reduces triglycerides, total cholesterol, increases the HDL cholesterol, and reduces the glycosylated hemoglobin. And products are accepted by the diabetic consumer. That is more important. If the products are prepared and they are not accepted by the target group, then there is no meaning for these for the development of the product. But these products are accepted by the diabetic consumers. Then coming to supplementary foods, these are there is an increment in the height by feeding these mixes or porridges. There is a improvement in the height, 4% increment in the height and 12% increment in the weight. This is done on the children of 8 to 15 months old, and over control there is an increment of 2.5 and 7% respectively with the height and weight. Then these are the protein energy dense mixes developed by including the ragi or finger millet and jowar with other ingredients. And there are two products developed. One is sweet and the other one is savory. And the sweet can be converted into a number of products, maybe like laddu or khachikai or barfi and things like that, which can be, which will facilitate the change in the diversity in the day-to-day consumption. And these are the savory products which can be prepared with this mix, that is nippatu, chakli, kodbade, and so many things can be prepared. And coming to sport-age food, has ideal protein under NIP project, and it enhances the endurance capacity and performance of athletes. It is useful for community as a protein and energy supplement. It provides 376 kilocalories of energy and 14 grams of protein, which provides 5 milligrams of calcium, 262 milligrams of calcium and 5 milligrams of iron. But the advantage here is this consumption of this food will improve for 90 days. It improved the hemoglobin status and physical fitness of the sports people. Endurance capacity was enhanced with this product, and the product was highly acceptable by the sports people. And then these are the pop sorghum health mixes, health foods. The pop sorghum was converted into meaty flavored, seasoned, and then health food, popped health food was developed, and this was developed to combat anemia among the adolescent girls. Then pop sorghum health mix, it contained 10.5 grams of protein and then 1.9, approximately 2 grams of crude fiber and 387 kilocalories of energy. Then this is AKJ1 red sorghum upma mix. Red sorghum, I just want to tell you, we know that sorghum will be vitable for the preparation of roti. But it is rich in fiber, antioxidants and minerals. And another thing is it is gluten-free totally. So this was converted into upma mix. Upma mix was prepared and which provided these nutrients like around 10 grams of protein, 144 kilocalories of energy and other thing. So this is the particle size of the semolina was standardized and then it was prepared. Then cookies were prepared. The specialty of these cookies is they are 100% sorghum. The particle size was standardized and then it was prepared. It doesn't contain any refined wheat flour or any kind of wheat. So it is gluten-free and it is most suitable for the people suffering from IBS and celiac disease. So the cookies, they are rich in micronutrient, fiber and antioxidants. So that can be one of the products. Then this is little millet cookies. They are ready to eat, nutritious, low trans fat, high fiber, high satiety value. That is more important, and they are suitable for children. This is the protocol of the preparation of cookies. Then breakfast muffins prepared with foxtail millet, and then ragi or finger millet, then finger millet cookies and finger millet bread. Then novel foods, some of the novel foods were tried, like flakes, RTC and RTE flakes were tried, noodles and pasta were tried. Then novel convenience foods, this is the millet noodles, then RTC flakes, ready to cook flakes, just like rice flakes, ready to eat flakes of barnyard millet, cookies of barnyard millet, and this is the little millet ready to cook flakes. These are all the ready to cook flakes of different millets, proso, barnyard, little millet, foxtail millet, and finger millet. They are all rich in iron, so they help in combating anemia, and they are also having high fiber content. High fiber content compared to corn and wheat flakes. This is the sensory evaluation of these RTC flakes. We can see that little millet flakes are highly acceptable, followed by foxtail millet and proso millet flakes. This is the flakes of red sorghum. Red sorghum flakes compared to white sorghum flakes. Red sorghum flakes are thin and they are highly accepted, just like flakes when they are seasoned. So they can be converted into, they were converted into many products like snack bar. One snack bar, that is breakfast snack bar, one bar will suffice on travel. So when person is traveling, one bar is enough, and this is rich in antioxidants and other nutrients also. It is good amount of fiber. These snack bars are prepared of different types. One is high energy, high protein snack bar, sorghum snack bar. That's a red sorghum snack bar. And other one is low glycemic index sorghum snack bar. These products were developed under Ministry of Food Processing Industries project by Dr. Hemlata and her group. And these are some of the products developed under the niche area of excellence. We have a project on millets. And under that project, these are the podo millet pasta, which is ready for commercialization. The product is ready for commercialization, and some of these technologies have been purchased already. This is pasta, and this is beetroot pasta, podo millet pasta with beetroot enhanced. And some instant mixes were developed under the niche area of excellence project, which is going on. This is barnyard millet dosa mix, then idli mix, barnyard millet idli mix, and then barnyard millet custard powder mix, then proso millet dosa mix, and proso millet pudding mix. This is proso millet chakuli or whatever is called in different name. And this is podo millet low fiber laddu, which is more suitable for the preschool children and children suffering from protein energy malnutrition, because this provides a good amount of protein, that is 16.38 grams of protein per 100 gram, and a good amount of calories also, 350 kilocalories. And then this is high fiber thalipattu prepared with podo millet high fiber fraction. And then this is the laddu prepared with brown top millet. And these are the millet snacks prepared, like little millet chakli, little millet lipattu, then foxtail millet laddu, and proso millet chakli. And then these are the products which have been commercialized already. The besan laddu, then preparation in the large scale. And then this is another sweet which is common here, traditional sweet. Then papads of millets, chakli of millet, little millet lipattu. And then this is little millet lipattu prepared in large scale and sold. So with all these products, what I've shown, developed in the department, there are some constraints or points to be considered to enhance the consumption of these millets. But still, with all these, the millets are not consumed by everybody. The people who were consuming millets earlier, they have stopped consuming. The main reason is availability and continuous supply of nutricereals. The thing is, many of the industries are ready to purchase the technologies, but the availability of the raw material is a problem. So the continuous supply of these nutricereals is very important. So also processing and storage of the grains. Then there is a need to develop region-specific acceptable recipes at the commercial level. And problem of high fiber intake, especially for children, will affect the mineral absorption. This has to be looked into. When we are preparing the food for children, we have to look into the fiber content, and fiber has to be reduced. Then the certain issues which have to be for solutions which we can think of. We can encourage the farmers for cultivation of nutricereal, but only when we give them assurance that the produce is purchased, then only they'll be ready to cultivate. So there is a need for direct procurement from the farmers, that is contract farming by the industries, and then inclusion of region-specific millets. Like in that particular region, whichever millet is common, that has to be included in the in different products. Maybe initially small quantities as a snack food or health drinks, and then it can be increased. So installation of destoning machine or grading machine, because one of the major hitch in consumption of millets is the processing of millets. Nowadays, of course, we are getting processed clean millets, but destoning is a big problem. So destoning machine, this unit is established by our own project, one of the projects by CEDA, that is Canadian project. So in this project, this unit was established in nearby village, and the person is processing quintals, 20 to 30 quintals every day, and he is selling it. So this destoning machine, after establishing this, availability of the grains has increased, and millet processing unit, which has been established, has helped to enhance the of millet. It provides food security, increases the entrepreneurship, augments the millet consumption, and also improves the health and welfare of the population. So with this, I would like to conclude that everyone should eat millets every day and stay healthy. So thank you for listening patiently. And before I conclude, I just want to thank the Padmavati University, Women's University, for giving me this opportunity. Thank you so much.
Thank you, madam. Thank you for your knowledge and informative, valuable information, madam. Any queries from the participants?
I'm sure they'll have a lot of questions. You can communicate with me later also. If you like, if you get any questions later, you can also contact me later. Dr. Sirisha can share my email ID or phone number. You can contact me later also, all the participants.
Yes, madam. Very good research, madam. Almost you covered all the millets. You developed lots of products from the millets, and you tasted all that one. It's a nice experience, madam.
Thank you. Thank you. It's not my own effort. It is the effort of the whole department. So I am thankful to the whole department. The department has permitted me to use the data for a presentation. So I'm thankful to the department actually. So I'm part of this project. Like we had initially we had a NATP project, that is technology, national technology project, and then innovative project. Now we have niche area of excellence. That is another World Bank funded project, which will be concluded shortly. So I'm part of this project. I'm not the main investigator, but I'm part of this project.
Yes. Madam, this all equipment is available, flaking machines, all these things are available in your institute?
Actually, flaking machine is right now not functioning, but we got it done in nearby place, that is Vijaypura. And there was a flaking unit, so we got it done there through them. And later we had one instrument established, but it is not functioning right now.
Okay, madam.
Yeah.
Madam, can you give us permission for students visit your university, madam?
Definitely, they can visit anytime. They can just drop a letter to our dean. So because now I'm retired now, so I can only tell you, you can drop a letter to the dean and they will permit you, definitely.
Thank you, madam.
Any time you can go and visit.
Good morning, ma'am.
Hello.
I have a query?
Huh?
Hello, madam. This is Jyoti, Professor Jyoti Pushpabharati
Hi,
garu.
Jyoti.
Good morning.
Hello, Jyoti
Good
madam.
morning, everybody.
Good morning, Jyoti madam.
Madam,
Huh.
making rotis with these millets is difficult. It is breaking. Edges breaking will be there.
Yeah.
So, can you suggest any technology to improve that?
Yes,
Easily, like wheat, we
actually,
cannot prepare into a soft consistency.
right. Yes, very true, madam.
Millets,
Because
no?
it is not having gluten, no. So
Yeah, that's right.
what we do is, madam, for jawar roti also. Hello, ma'am, for jawar
What?
roti also, what we do is, we first gelatinize the flour. Jyoti madam, I hope you are able to hear me.
Yeah.
Yeah, ma'am, we first gelatinize the flour. And then once we gelatinize, it becomes sticky. So that can be used for rolling. It can be rolled once you gelatinize.
I heard that in Karnataka, they are selling the flour which can be easily rolled like this. They are pre-gelatinized products, powder?
No, ma'am, no, ma'am. Pre-gelatinized is not available, but the flour is available, which you have to gelatinize it. Part of the flour is gelatinized and dry flour is mixed. If it is fresh, like if it is old flour, then what we do is we gelatinize the whole mass, just like rice flour we gelatinize for some products, no, ma'am. That way.
Not gelatinizing ourselves, so the powder which is prepared by the Shakti groups, I think. Shakti group, they are selling such a product, powder, with all millets. I think they are doing some technology. They are using some technology to roll it like that.
Okay.
Like a wheat flour. It can be rolled like anything, just mixing with the water and rolling. So some technology they are using, I don't know. It's a business secret, no? But they are selling like that. So I think they are using some pre-gelatinization or something, I think so.
Maybe, but I'm not aware of this, madam. I'll just check.
Shakti groups, I think. Shakti groups is selling that product.
Okay. Lot of products are available. The millet mixes are available. So maybe it is one of them. I'll just check it out, madam, if I get it, because I'm not aware of
Thank you. Thank you, ma'am. Thank
you. Thank you.
Madam, one of the participant asked in the chat box, madam.
Okay.
One doubt.
Okay, this question, I have read an article on eating millets daily will increase the calcium content and may have indigestion. Okay, I'm so sorry, I forgot to tell you about this. Actually, the people who are not used to millet should not consume it every day. Because if you start consuming millets regularly, maybe initially you may have indigestion and loose motion and things like that. So slowly you should start increasing the consumption. You can't consume because now people are I mean, thinking of advantages, people are consuming millets, they are becoming mad about millets and consuming it in every meal. So I don't recommend that initially. So those who are used to consuming millets, they can consume, but those who do not consume, they don't know about it. You start consuming from small quantity, maybe a day, one meal a day, one product in a meal, and then slowly you can increase. So definitely it will lead to indigestion in old people. The calcium availability, the increase is there, of course, but the availability is a question because of high fiber. For calcium availability, mineral availability is reduced because of high fiber. That is there. So it has to be balanced, of course.
Any other doubts, ma, from the participants? Otherwise, we conclude this session.
Madam, another question is there. Please share about anti-nutrient contents in the millets.
Anti-nutrients are quite good actually. The study what we have done, it varies from one millet to another millet and depends on the processing also. So I have the data. I can share the data individually. Whoever wants it, you can contact me. I'll share the data with you. Okay. I hope I have answered. Dr. Sirisha?
Yes, madam. Yes.
Okay. You can give my number or email ID. I will share the data.
Sure, ma'am. Thank you.
Yeah.
Niharika, let us conclude, I think.
Okay, madam. Okay. Thank you so much, Professor Pushpa Bharati, madam, for accepting our invitation, giving a nice lecture about millets and its importance. You are enlightened different types of major and minor millets and their developed products, traditional foods, sweets, snack foods, and sports foods. These are all very helpful for all the people. Now the COVID-19 pandemic situation, people are aware of their health. Regarding lifestyle changes are also now we can see, madam. For that purpose, you are giving very valuable suggestions and information, madam. Thank you so much, madam.
It was my pleasure. You have included me, so I'm very thankful to the university. And it was my pleasure to share anything on millets because we have worked so much on millets. No? So I just, whatever we have done, I have just shared with you. Thank you so much.
Thank you, ma'am.
Thank you, ma'am. In future also, we are expecting your help like this.
Definitely. I'll be there. As long as I am there, I'll be there with the university. No issue.
Madam, can you share your PPT with our conference mail, madam?
Yeah, I'll do.
Thank you, madam. Okay. Thank you.
Ma'am?
Yeah.
Ma'am, thank you for the information. Ma'am, I have a doubt. Ma'am, how about giving millets for thyroid patients, ma'am? Because there was one topic that saying that millets are against thyroid hormone, ma'am. They do have the same action as cabbage and cauliflower, ma'am, as goitrogen, ma'am.
No, I don't think they have any goitrogenic factors. I'm not aware of this actually, but I don't think it will have any effect because they are not sulfur-containing compounds. Many of these are not sulfur-containing compounds. Like cabbage, cauliflower, or even beef. They are all goitrogenic substances. So they are not. I don't know. I'm not come across any of these things, to be frank. But I can just go through. This is something you told me to look into now. I can look into this information. I'll just do that.
One request, madam. I request Aruna ma'am and Manjula ma'am, all of you turn on your cameras, ma'am. We will take one virtual photo here.
and share it.
Sure, ma'am.
Yeah.
Aruna, ma'am.
Aruna madam is there.
But ma'am, photo is not appearing. Bindu,
No,
ma'am.
she is not switched on the video, I think.
Switch.
Jyoti madam
Bindu,
is
ma'am, switch on your video. And madam, other department faculty members, please, anybody is there, switch on your videos. We will take one virtual photograph.
Blouse.
Aruna, ma'am. Aruna, ma'am.
On చేస్తున్నారు మేడం వీడియో.
But
No, I don't think. Audio is on. No.
మా ఒక్కరు కాల్ చేయరా మేడం కి?
Ma'am, I am taking photos, ma'am. Arna is displaying my video.
Okay. Thank you, ma'am. Okay. Madam, any queries or any doubts? Now we will break for tea, madam, virtual tea. Yeah. Thank you, ma'am. After tea break, we will join for next session. Next speaker, Manjula ma'am, is already joined. Ma'am, after 10 minutes, we will join, ma'am.
Yes. I'll leave this and join with another system.
Yes, madam. Thank you. Thank you, ma'am.
Fine. Thank you so much. Bye, Jyoti ma'am.
Bye, ma'am. Bye.
Bye.
See you sometime. Talk to you sometime.
See you afterwards. After break. See you after break.
Huh?
Jyoti madam, good morning.
Good morning, ma.
Hi, Jyoti garu.
Hi, Rajini.
Good morning, Jyoti ma'am.
Morning, ma.
Good morning, madam.
Good morning.
How are you, ma'am?
Fine, thank you.
Enjoying with kids, ma'am.
Huh?
Kids తో ఎంజాయ్ చేస్తున్నారా అంటున్నా.
Full.
వెరీ చిన్నమా మేడం?
వేసుకోవడానికి వెళ్ళాలా 1 o'clock కి.
ఓహో.
షెడ్యూల్ చేసుకున్నాను.
ఓకే. అందరూ బాగున్నారా మేడం? మేడం కొంచెం బెటర్ మేడం.
ఇంకా వాయిస్ అట్లా ఉన్నట్లుంది.
వాయిస్ కొంచెం సెట్ అవ్వలేదు, ఎలర్జీ కొంచెం తగ్గింది.
మ్యామ్ గుడ్ మార్నింగ్ మ్యామ్. జ్యోతి మ్యామ్, రజనక్క, బిందు.
హాయ్ మంజు.
గుడ్ మార్నింగ్ మంజు.
గుడ్ మార్నింగ్. జ్యోతి మ్యామ్ ఏం మ్యామ్ సిగ్గా ఉన్నారు?
మేడం బూస్టర్ జ్యూస్ వెళ్తారు.
నిన్న బాగా ఫ్రెష్ గా ఉన్నారా మేడం?
ఈ రోజు డల్ గా సిక్ గా ఉన్నారు మేడం.
బూస్టర్ డోస్ కి వెళ్తారు కదా అందుకే.
అంటే వెళ్తున్నారా, వెళ్ళేసి వచ్చారా?
వెళ్తారు.
వెళ్ళాలి 1 o'clock కి షెడ్యూల్ చేసుకున్నా.
వెళ్ళే ముందు సిక్ అయిపోయారు మేడం.
హాయ్ ఎవరక్కడ? హలో
ఆంటీ అనాలా?
ఆంటీ లేదురా, కొంతమంది అమ్మమ్మలు కూడా ఇక్కడ. ఏం మంజు ఎలా ఉన్నావు?
అక్క చెప్పండి అక్క.
ఎలా ఉన్నావు మంజు?
బాగున్నా అక్క. మీరంతా బిజీ వర్కింగ్.
చిట్టి
ఆన్లైన్ క్లాస్
మీకు కూడా ఆన్లైన్?
హాయ్ చిట్టి, ఎలా ఉన్నావు? హలో చిట్టి, బాగున్నావా?
అవునులే మంజు.
తమ్ముడు బాగున్నాడా?
ఇప్పుడే మీరు
బాగున్నారా? నాకు జ్వరం చిట్టి, ఇంట్లో బాగాలేదు.
ఏదో అడ్మిషన్స్ మాకు జరుగుతా ఉన్నాయి కదా
మేడం. నేను సిక్.
ఏం అడ్మిషన్స్ అక్క?
ఇంటిగ్రేటెడ్.
పాప బాగుంది మేడం. నేను
యాక్చువల్లీ ఫస్ట్ సెమిస్టర్ వాళ్ళు వస్తారేమో కదా నెక్స్ట్ వీక్.
పెద్ద అయిపోయిందా?
రెడీగా ఉన్నారు
పెద్ద
మంజు.
అయిపోయింది
ఎప్పుడు
మేడం.
రమ్మంటారు అప్పుడు రావాలి వాళ్ళు.
బాగుంది మేడం.
పృథ్వీ ఎట్లున్నాడు మంజు?
బాగున్నాడక్క. పిల్లలు పంపించేస్తారు.
బాగున్నావా మంజు?
మేడం బాగున్నాను మేడం. బాగున్నాను మేడం. పృథ్వీ ఫైనల్ సెమిస్టర్ మేడం. పిల్లలు చాలా పెద్దవాళ్ళు అయిపోయినారు. వీళ్ళు పిల్లలు అబ్బాయిలు అంత పెద్దవాళ్ళు అయిపోయారు. మొన్న పుట్టినట్టున్నారు.
అదే
పాప వెళ్ళిపోయిందా బెంగళూరుకి?
వెళ్ళిపోయింది మంజు.
మీరు ఫ్రీ.
తప్పు చేయకండి, మాట్లాడుతున్నా కదా ఆంటీ వాళ్ళతో. ఏం మధ్య ఈ పిక్ ఏదో పెట్టుకున్నావు, ఫేస్ చూపించకుండా?
మేడం ఎలాగో మళ్ళీ తర్వాత ఆన్ చేయాలి కదా మేడం. అంటే ఈ రోజు కొంచెం క్లైమేట్ చేంజ్ అయింది మ్యామ్.
అవును,
మాకు మా ఏరియాలో స్లైట్ గా వర్షం డిజ్లింగ్ లాగా పడుతోంది అండ్ గాలి ఎక్కువ ఉంది మ్యామ్. అందుకని భయపడుతున్నాను.
ఇంటర్నెట్ భలే డిస్టర్బెన్స్ ఉంది.
అవుతుందేమో.
2 days బ్యాక్ నాకు అట్లే ఉంది జ్యోతి గారు. నేషనల్ గర్ల్ చైల్డ్ చేసినాము. మధ్య మధ్యలో ఎంత టెన్షన్ ఉండిందో, ఎక్కడ పోతుందో, ఎట్లా నేను జాయిన్
బాగానే ఉన్నట్టుంది కదా.
కొద్దిసేపు జాయిన్ అవ్వలేకపోయినా నేనైతే మొన్న. మళ్ళా జాయిన్ అయినా.
ఈ రోజు కూడా బాగా విపరీతంగా గాలి ఉంది కదా, డిస్టర్బెన్స్ వస్తుంది.
నేను తిరుపతి నుంచి వచ్చేసినప్పుడు అంతా
మేడం
తిరుపతిలో లేరా ఇప్పుడు?
హైదరాబాద్ లో ఉన్నా. తిరుపతిలో ఉన్నన్ని రోజులు ఎండే. నేను వచ్చేసాను వర్షాలే. రోజు ఏ రోజు చూసినా వర్షం అంటున్నారు.
మేడం యూఆర్ ఆన్ మ్యూట్ అనుకుంటా, వినిపించట్లేదు అరుణ మేడం.
అరుణ అన్మ్యూట్.
బిందు ఎలా ఉన్నావు బిందు?
సిక్కుగున్నా మేడం కొంచెం. ఇంకా
బిందుకు అలర్జీ వచ్చింది, చాలా అవస్థ పడుతుంది మేడం.
గుడ్ మార్నింగ్.
గుడ్ మార్నింగ్ మంజు. గుడ్ మార్నింగ్.
ఎలా ఉన్నారు మేడం?
ఏం ఎలర్జీ బిందు?
డ్రగ్ ఎలర్జీ మేడం. డ్రగ్ ఎలర్జీ.
డ్రగ్ ఎలర్జీయా? ఏమన్నారు బిందు డాక్టర్? నాకు పర్వాలేదు మేడం, ఉంది ఇంకా థ్రోట్ ఇన్ఫెక్షన్ ఇంకా తగ్గడంలా. అంటే థ్రోట్ అంటే వాయిస్ మారిపోయింది చూడండి. అవస్థలు పడుతున్నాం. ఎవరిని అడిగినా కోవిడ్ అంటున్నారా మా తల్లి? నేనే పర్లేదు అనుకుంటా.
జ్యోతి గారు టచ్ వుడ్, వీళ్ళందరికన్నా నేనే బెటర్.
రజనీ ఒకటే కొంచెం ఆరోగ్యంగా ఉంది ఇప్పుడు. 3 ఎపిసోడ్లుగా అయిపోయినాయి, అయినా బానే ఉన్నా నేను. కాబట్టి మీ అందరికంటే నేనే హెల్దీ అన్నమాట.
మీతో పాటు నేను కూడా.
షేర్ యువర్ సీక్రెట్స్ మేడం. మీ సీక్రెట్స్ షేర్ చేయండి మాతో. మాకు ఇమ్యూనిటీ చాలా లో అయిపోయింది.
ఆల్వేస్ గుడ్ సోషల్ నెట్వర్క్స్.
అంతేనంటారా?
ట్రై టు హ్యావ్ గుడ్ సోషల్ నెట్వర్క్స్. అదే హెల్త్,
హ్యాపీనెస్ ఆల్వేస్.
మంజు బాగున్నారా? మంజు
మేడం బాగున్నా మేడం. ఏం మేడం ఫీవర్ మీకు కూడా?
నాకు కూడా కోవిడ్ అటాక్ అయ్యి, నాకు చిన్నుకి. ఏంది బిందు నీకు కూడా కోవిడా?
మేడం నాకు కోవిడ్ కాదు, నాకు ఫుడ్ అలర్జీ, డ్రగ్ అలర్జీ మేడం. నాకు ఊరు వెళ్ళడం వల్ల వైరల్ ఫీవర్ స్టార్ట్ అయింది. అక్కడ నుండి ఆ మెడిసిన్స్ ఎక్కువ అయిపోయినాయి అంట, అధికంగా వేసుకున్నానంట. సో నాకు ఒళ్ళంతా ఎలర్జీ అనమాట. ఇచ్చింగ్ కూడాను. ఇది వరకు నాకు ఫుడ్ ఎలర్జీ కూడా ఉంది కదా మేడం, ఇప్పుడు డ్రగ్ ఎలర్జీ కూడా వచ్చింది.
డ్రగ్ అలర్జీతో ఫుడ్ ఎలర్జీ
అన్ని ఫుల్.
మంజు
మేడం చెప్పండి మేడం.
మంజు నేను
వీడియో ఆన్ చేసుకోండి మంజు.
నేను పూర్తిగా ఉండలేనేమో మంజు నీ టాక్ లో.
సరేకా
నేను అంటే యూనివర్సిటీకి వెళ్ళాలా ఏదో రమ్మన్నారు అందుకు సేపు ఉంటాను.
ఆన్లైన్ అంటే ఇంక అందరం ఇంట్లో ఉండిపోవాల్సిందే.
అంటే ఆల్మోస్ట్ ఉంటున్నాం అవసరమైతే తప్పితే. ఈరోజు క్యాంపస్ లో ఏమైందంటే NMBP మెడిసినల్ ప్లాంట్స్ మీద సెమినార్ అది వాళ్ళు ఆఫ్లైన్ పెట్టినారు. అక్కడ ఒకతను వస్తాడు, ఆయన్ని మీట్ అవ్వమన్నారు, అందుకని వెళ్ళాల్సి ఉంటారు.
ఓకే, అదే మనకి యాక్చువల్లీ నెట్వర్క్ ఉండదు కాబట్టి యూనివర్సిటీలో అన్ని ఆన్లైన్ ప్రోగ్రామ్స్ ఉంటే మనం ఇంట్లో ఉండిపోవాల్సి వస్తుంది.
అవును అవును.
Sure.
Welcome back to you all. Beulah?
Yes, ma'am.
Okay. Who are sharing the screen, ma? Niharika?
Myself, I will share, ma'am.
Okay. Just a minute, I will start the recording.
Okay.
Welcome back to you all. Good morning, Manjula madam. I welcome Beulah to introduce today's speaker, Manjula ma'am. Welcome, Beulah.
Thank you, ma'am. So I would like to introduce today's guest speaker, Dr. Manjula Kola. Presently, she is working as Associate Professor, Department of Home Science, Sri Venkateshwara University. So madam's specialization is in food science and nutrition and food technology. Coming to her educational qualification, she did her M.Sc and PhD in food science and nutrition from Sri Padmavati Women's University, Tirupati. And madam has 24 years of teaching and research experience. Madam received different awards. The list is here. She received Professor Sarveshwara Endowment Award and Best Outstanding Academic International Award in 2020. Madam also received recently the Best received Andhra Pradesh State Scientist Award in Food Technology by Andhra Pradesh Council of Science and Technology in 2021. She also awarded with Adarsh Vidya Saraswati Rashtriya Puraskar in 2021. She also received Mahila Shiromani Award in the field of education. Dr. Manjula Madam published 55 research papers in reputed journals. She published 3 books and 11 book chapters. Madam has 3 professional patents. She guided several students, PhD 5 students, MPhil 1 student, and 60 PG students. Madam handled 3 projects, research projects, which includes the major research project funded by UGC with entitled Development of Functional Foods with Potential Sources and Transfer of Technology to the Community for Health Promotion. And she also received, developed 3 e-learning modules for UGC e-Patashala. Madam, as a resource person, developed 20 invited talks in different organizations. She attended different international seminars, conferences, and workshops. She performed various administrative positions and active member in different professional and research bodies. Currently, she is serving as an editorial board member for Journal of Sports, Nutrition and Therapy, and current research in nutrition and food science, and International Journal of Multidisciplinary Research. So now I request Dr. K. Manjula ma'am to deliver her talk on probiotics, prebiotics, and symbiotics as a functional food to address lifestyle disorders. I welcome you, ma'am. Now the session is over to Dr. K. Manjula ma'am.
Thank you very much, Beulah, for a nice introduction of mine.
Thank you, ma'am.
The organizers of the international conference, Dr. Sirisha, Professor Arna ma'am, Professor Jyoti ma'am, Professor Rajni ma'am, and my dear friend, Professor Bindu, and very young and dynamic organizing committee members and the participants. Very good morning to all of you. I'll share my screen once again. Beulah, is it visible, ma? Is my screen visible?
Yes,
ma'am. Yes, ma'am.
Thank you. So, dear participant, I just start my session with a self question, because it's very important to question ourselves with regarding to this particular session, question, self question. So we know that everybody, all of us are want to be healthy, want to be fit, and disease prevention, because free from diseases throughout our lifestyle, isn't it? It's right, because many of us are thinking towards achieving a sustainable health throughout our lifestyle, throughout our life. So if it is yes, if you want to be like that, we have to question ourselves that whether we are leading a healthy lifestyle or not. Because it's very, very important that why I am starting my session with this self question, because number of research studies in medical profession as well as nutritional sciences, they are revealing that there is a strong relationship between the way that we are living, that is our lifestyle, with chronic diseases occurrence and progression, with special reference to lifestyle diseases. So if you see the latest reports of scientific literature, from the scientific literature, it is known that the world population, among the world population, 75% are claimed to be unhealthy lifestyles. And if you see the Indian perspective, over 61% of all deaths in India are attributed to our lifestyle. It is according to the records of Center for Science and Environment. So lifestyle diseases are getting increasingly common across the age groups, irrespective of the age. It is very common among the children, and it is very common among the youngsters and in old age people too. So the diseases, what are lifestyle diseases actually? The diseases which arise from the way that we are living are lifestyle diseases. Sometimes we are also called them as non-communicable diseases. If you see the reason, why? What is the reason behind that? What is the reason that much progression in the lifestyle diseases in recent years? Again, if we question ourselves, the answer is the simple routines we set for ourselves on a daily basis are the reason. If you see the complexity in the etiology of lifestyle diseases, we see it's really a kind of complex etiology in lifestyle diseases. Whenever you are checking with like any lifestyle disease, the factors are quite different from one to one. And the one of the factor, which is a prime and very important contributor for unhealthy lifestyle, which leads to many chronic inflammations and diseases, or nutrition transition. So we as a nutritionist and food technologist, we know very well that since previous years, since long time, we are slowly progressing. We are slowly shifting from consumption of traditional foods to more processed foods. Again, there are many reasons behind that, because urbanization, industrialization, and globalization, and women empowerment and employment. So these are all various reasons for more choosing convenient foods, which are more processed ones. And we are neglecting about the more natural, fresh, and traditional foods, which are nutrient dense and more healthy. So this is one of the reasons for more occurrence of lifestyle diseases in recent years. And the other factors, everybody know that increasing inactivity. So because of technological advancement, we are all depending, we are sitting in front of the computers and we are depending more on the equipment rather than using our manpower and energy for performing our day-to-day tasks is again one of the reasons for progression of these lifestyle diseases among people. And stressful situations, the stress is one of the other factor, and lack of sleep. This is very, very important factor. Majority of the health professionals are concentrating in recent years with regard to lack of sleep, because we know that during sleep only our system repairs. So major detoxification process has been happening during our sleeping hours. If there is a lack of sleep, what happens? Everything is disturbed. Our biological clock disturbed. All circadian rhythms are changed. So again, that is lead to lifestyle diseases. And lack of time and habits like smoking and alcohol and the frequent consumption of drugs, particularly antibiotics, and very important factor that are major responsible for lifestyle diseases among youngsters is the faulty diet and fasting regimen without expert advice. So this is the one that is observed in recent years. The major reason again is the social media, and people are blindly following the social media, and they are following faulty diet and fasting regimes for mainly for weight reduction and for fitness, etc. So these fasting regimen, and in recent years that we are seeing that intermittent fasting came into vogue and becoming very famous among youngsters for weight reduction and the fitness. Of course, as a nutritionist, that we can agree that intermittent fasting is very, very good and very best approach for weight reduction, but it has to be followed under the guidance and under the supervision of either the dietitian, nutritionist, or a health professional, because there are certain kind of principles and regulations are there to follow the intermittent fasting. And these are all other major factors in the etiology, which leads to unhealthy lifestyle of the individual, ultimately leads to the lifestyle disorders. Then what is solution? So if for every problem, we have to seek for a solution. So if I want to suggest a solution, as a nutritionist, I can say that food is a medicine, of course, but it is not the current, the recent mantra, modern mantra of nutritionist and dietitian. This particular quote is coming from generations. So everybody know about Hippocrates. He is the Greek physician. He gave a lot of quotes with regard to the prevention of the disease, especially with regard to food. So one of the best quotation with regard to food of Hippocrates is, let food be the medicine and medicine be the food. So everybody familiar with that particular quote. So according to this mantra, according to these quotation, we are consuming a very healthy and balanced diet to keep our fitness and to keep our health in a right condition. to free from the diseases. So is consumption of healthy food or is the consumption of balanced food alone can be a strategy for prevention of the disease? If you question again, so no. Why? Because the consumption of a healthy food or the consumption of a balanced diet is really a holistic act. Because instead of only considering about what we are eating, we have to consider what we are eating along with how we are eating and when we are eating and how we are preparing that particular food for consumption. These are all has to be considered while we are consuming a very healthy and balanced diet. So a lot of wisdom has to be required for the individual while planning a balanced diet for yourself to keep your health and fitness in a right way. So we know that we have a number of microbial flora in our gastrointestinal tract. This gut bacterial flora, as well as the mucosal lining integrity of our gut, is strongly connected with chronic inflammatory conditions. So when you see the etiology of chronic diseases or any degenerative non-communicable diseases in a textbook, in your dietetic textbook or your medical textbook, in the etiology, the previous textbooks in the etiology, so the etiology is different when compared to the latest editions. Because according to the generations, the etiology of the disease has been changing. And here in recent years, the medical research are more concentrating about gastrointestinal tract and the microbiota which is present in the gastrointestinal tract and the role of that microbiota in the occurrence and progression of diseases, many diseases, and with especially the lifestyle diseases. So understand that our digestive system and its ability to metabolize various foods, that is a key factor in development of the functional foods. When we are developing functional foods, the food technologists and the researchers and nutrition researchers or nutritionists has to be very thorough knowledge, very in-depth knowledge with regard to the functional ingredients that we are incorporating in the designing and development of the functional foods. And what is the composition of the functional ingredient and what is the interaction between the ingredients that are there that we are putting into the food matrix and impact of the processing technologies when we are developing the food product. These are all very, very important factors, important points that to be considered while we are developing the functional food products. And with regard to the today's session on probiotic, prebiotic, and symbiotic, again, I am giving one more quotation of Hippocrates that he said that all diseases begins in the gut. But it's very, very important. It's very much true in case of all the disorders, particularly in recent years. So what we have to do to prevent the disease or to treat the disease? So you have to heal your gut first. You have to heal your bowel first. Take care of your bowel, take care of your gut first while you are entering into the developing the nutritional strategy and following any implementation of nutrition or any taking any supplements, etc. First heal your gut first. And this is a very important point that has to be considered. And we are talking about the gut and gut microbiota. We just have a brief look into the gut microbiota. What exactly it is, how it leads to occurrence of many diseases with special reference to lifestyle. So you know that we have more than 100 trillion microorganisms colonize the gastrointestinal tract. They are playing a vital role in efficient nutrient absorption as well as maintaining the human health. Therefore, what we have to do is optimum balance is required between the intestinal microbiota and the host. This is very, very important point. So in some circumstances like frequent infections and aging and environmental pollution, etc., this balance may be changed. So this may cause gut disturbances, which leads to chronic disorders. So knowledge of gut microflora and its interactions may help in development of strategies, particularly in functional food development. And let's see a look into gut microbiota. But we have a certain kind of knowledge with regard to the microbiota. So any normal individuals, generally, most of the individuals, the composition of human gut microbiota is 65% is occupied with firmicutes, and 30% is bacteroidetes, and remaining 5% with other kind of microorganisms. There are various habitats are there in the human system, including the gastrointestinal tract, where these particular microbiota is located and performing various physiological functions to protect us from environmental pollution, toxins, and other things, so that we are sustaining with better health. So we had to know about the important vital functions of gut microbiota. Because when we are developing a prebiotic or a probiotic or both like symbiotic food product, we must know about what kind of microbiota is there already in our system and what are the vital function of that particular microbiota, where the imbalance is occurred, and what is the problem of the individual, because for what purpose that we are going to develop that particular product with probiotic or symbiotic product. So what is the function that we required? So these are all the various things that we have to keep in mind before itself when we are formulating a product, functional food product based on probiotic or symbiotic. Here you see that the microflora in the gastrointestinal tract have many vital functions. So first one, very important function, it is role in digestion. So we already discussed that taking very good diet with a balanced kind of nutrients is not only alone is not only help us to prevent from the diseases, because keeping our physiological system, keeping our gut health is very important, because whatever we consuming, that has to be digested properly, that has to be absorbed into the system properly, and that has to be utilized by the body properly to perform the various physiological functions. So the microflora in the gut, it helps in digestion. So how it helps in digestion is, it is involved in the metabolism of carbohydrates, proteins, and fats, and it yields small chain fatty acids after digestion. These small chain fatty acids such as acetate, propionate, and butyrate has a lot of significance in prevention of the diseases. So how it acts in the health? What is the role of it? If you see acetate, it helps in appetite regulation, and it is one of the vital ingredient that are using in various product development for weight loss. So propionate is another small chain fatty acid. It also call it as satiety molecules. So it just to switch off the hunger in the individual. And butyrate, this is a very important component. It functions and it promotes the apoptosis. So particularly apoptosis in malignant epithelial cells. So that helps in reducing the risk of colon cancer or the bowel cancer. So these are all the various components that are released after digestion of food, which is converted into short chain fatty acids by the gut microbiota. So you might know the importance of gut microbiota in the system to maintain our health now. So apart from the role in digestion, the gut microbiota also perform a very vital function, that is vitamin biosynthesis. And we know that our tissues, our cells, they are not directly synthesize many of the vitamins which are essential for our body. So for that purpose, we are depending on diet and food intake and our various kinds of nutrients intake, as well as the biosynthesis of nutrients by the gut microbiota. These gut microbiota synthesize various vitamin B vitamins as well as vitamin K in the gastrointestinal tract. Particularly, this particular function is very, very important while you are designing a vegan foods, because you know that vegan foods are very familiar in recent years. Many of the people are turning into vegans. So many of the celebrities and sports people are also converting as vegans. So supplying the right kind of protein and sufficient vitamins, particularly vitamin B12, the vegan food, vegan diet is very, very difficult for a dietitian or the nutritionist. So here, with regard to vitamin B12, for instance, this is synthesized by our body, and gut microbiota is playing a major role in the synthesis of vitamin B12. So what happens if there is any disturbance occur, any disproportion occur in the synthesis of these vitamins in the body, and vitamin B12 is not available in vegetarian food. So what about the vitamin E, vitamin B12 requirements for vegans? That is again a very important question that we have to see. So microbiota is very, very important for vitamin biosynthesis in the gut. And the other vital function is xenobiotic metabolism and interaction. But the role of gut microbiota and xenobiotic metabolism and interaction, this particular area is still in infancy in research. You know what are xenobiotics? Xenobiotics are certain kind of chemicals which are coming from environmental pollution, toxins, and food additives, including preservatives and sweeteners, artificial sweeteners, flavorings that are added, that we are added into the food to enrich the quality of the food and give value addition to the food, etc. These particular chemicals, the gut microbiota is metabolized in the xenobiotics, and they excrete the unnecessary end products of these xenobiotics and keep our body in a health and condition. So this is again one more vital function of our gut microbiota. And other very, very important function is gut-brain axis and brain-gut microbiome axis. So what is it? It refers to the direct and indirect links and signaling between the enteric and central nervous system. So what happened? How the microbiota playing a role in this particular function is like, so we know that short chain fatty acids are released after digestion of the food. And these short chain fatty acids, they are able to stimulate the release of the hormones from neuroendocrine cells of the gut, which include peptide Y as well as glucagon-like peptide 1. So what are the functions of these two hormones, or very, very important functions that they are performing? The first one, peptide Y, which is a satiety hormone that reduces the urge to eat. And the other one is, which in addition to suppress the urge to eat, it promotes the satiety. So these hormones are very, very important, and this particular function is very helpful while we are developing functional foods targeting overweight and obesity. And the other important function is immune modulation. This particular functional property of gut microbiota have very familiar and becoming very crazy during COVID-19 pandemic. So it helps in immune modulation. So, you know, the infants, for instance, you know, the infants who are breastfed are having more immunity, that we know, more immunity when compared to the infants with the bottle fed. So the breast milk, we know, till past, you know, we are thinking that breast milk is very sterile in nature. So in recent research, so the findings of recent research stated that breast milk contain live microorganisms which are known as probiotics. So along with this, breast milk also contains fructones which are having prebiotic properties. So that's why breast milk is one of the source for both probiotics as well as the prebiotics. So that's why breastfed babies are very strong enough to various kinds of infections. So what happened if there is imbalance in the number and diversity of gut microbiota in gastrointestinal system? That is one particular condition occurred, which is known as dysbiosis. So when dysbiosis occurs, for the normal healthy individual, the composition of gut microbiota and the number and diversity of gut microbiota is in normal condition, and that is helpful for all the functions, and that's why we are in health. We are very healthy and fit. So when dysbiosis occurs, so everything was changed. The scenario was changed. Why dysbiosis occurs? It's because of the food diet. Food diet, the more processed ones, not natural ones. The diet which is low in fiber, the diet which is low in antioxidants. So a very poor, high in fat. These are these kind of diet is one of the reason. And stress and direct exposure to pathogenic organisms. Frequent exposure to toxins, environmental toxins and pollutants, and frequent infections. These are all one of the reasons. And antibiotic therapies. So when the person was frequently infected, the continuous uses of antibiotics are, they followed the continuous uses of antibiotics. These antibiotics kill most of the friendly bacteria and healthy bacteria in the gut. This is also one of the reasons for imbalance that is occurred in the number and the diversity of microbiota. And just now I told you that bottle-fed babies. So bottle-fed babies, the dysbiosis is there in that infant gut. So what are the consequences of this dysbiosis? If imbalance occurred in the composition of the gut microbiota, what happens? So if in the dysbiosis, loss of probiotic bacteria and microbial diversity occurs first. So it becomes very detrimental to the health of the individual. So this is a situation where there is a lot of demand was created since few years towards functional food. And consumers are more conscious with regard to the health, and they want to buy the functional food. They want to consume functional food, which are helpful for their system, which are helpful to promote their health. And here, the health industries, nutraceutical industries, and pharmaceutical industries, they are putting efforts for producing more foods, more functional foods with live bacteria and probiotic bacteria, and probiotic foods and probiotic food products. This probiotic, prebiotic, and symbiotics are well known in health food market and functional food market because this is one of the important category of functional foods. So what are probiotics? So, probiotics are the microorganisms that are beneficial effect on host intestinal microbial balance. But recently, the definition has been changed. The definition was changed by WHO with regards to the probiotic and prebiotic as well as symbiotic, because there is a lot of demand in the amongst consumer with regard to these particular foods, and food industries are manufacturing the functional food products with these particular organisms and marketing. So in this context, to keep the safety and to keep in mind the safety of the consumer health, so the WHO changed the definition of these probiotics recently. According to the WHO and FAO, probiotics are live microorganisms which, when administered in adequate amounts, confer a health benefit on the host. There are a lot of probiotic species are available, and with regard to the product development, particularly functional food product development, the most useful the most applicable probiotic species are lactic acid bacteria, bifidobacterial, which are most common types of microbes. But certain kinds of yeast and bacilli may also be used because more cereal-based and pulse-based probiotic foods are also came into way. And for the development of that particular purpose, certain kinds of yeast and bacilli are also used as probiotics in the development of functional foods. And the other term in our session is prebiotics. So prebiotics are mostly fibers that are non-digestible food ingredient and beneficially affect the host health by stimulating growth and activity of gut microflora. It helps facilitate the growth of gut microflora in the human system. So if you see the sources of prebiotics, there are a lot of sources of prebiotics are there. I already told you breast milk is one of the prebiotic. Why breast milk called as prebiotic? I gave the justification just now. And soybeans and inulin sources. Inulin sources are very frequently used prebiotic in many of the products. And unrefined oats, unrefined wheat, unrefined barley, or one of the excellent sources of prebiotic. And apart from that, non-digestible carbohydrates with particularly oligosaccharide family, or very, very important and the most beneficial prebiotics. Coming to symbiotics. So using prebiotic and probiotic in combination is generally we call it as symbiotic. But I told you that this WHO FAO changing the definitions of all these foods. So according to the FAO definition, the term symbiotic has to be used only if the net health benefit is synergistic. If the benefit is synergistic, then only we have to term symbiotic in our product label or in the promotion of our products. So how it works synergistically in the human system, like when symbiotic food is ingested, it is having both pre and probiotic, right? The probiotic, the prebiotic which is present in symbiotic, it enhances the survival of probiotic in the upper part of the gastrointestinal tract. I'm showing in that figure, where actually it functions, where actually the probiotic acts and where actually the prebiotic acts, how it acts synergistically. So the prebiotic acts at upper part of gastrointestinal tract, whereas probiotic works in the small intestine. Thus, it works synergistically in both the parts, both upper and lower part, both large intestine and small intestine. That's why we call it as symbiotics. Symbiotics generally work in two ways. First one, by improving the viability of the probiotics. This is very, very important thing that food industry is facing. One of the most challenging part while developing functional foods with probiotics is the viability of probiotics. So the symbiotics helps to improve the viability of probiotics. Apart from that, it is also works by delivering specific health benefits to the host. And there are there is a documentation, lot of research findings with regard to the therapeutic uses and the health benefits of all three, probiotic, prebiotic, and symbiotics. The most common documented benefits are it helpful in chronic diarrhea, it helpful in constipation. It is helpful in many of the degenerative diseases and non-communicable diseases like cardiovascular, diabetes, type 2 diabetes, and many types of cancers, particularly colon cancer and bowel cancers, all bowel cancers. And it is having anti-inflammatory effect, antibacterial activity, and antioxidant activity it have. And we just discussed that it synthesizes a few significant nutrients with special reference to vitamin B and vitamin K, and which is one of the very important and very significant property in developing vegan foods, vegan functional foods. And it alleviates the symptoms of allergies and respiratory and urinary tract infections. So lot of beneficial effects are on aging. So many geriatric foods, these particular probiotics and prebiotics has been incorporating in many geriatric foods that are developing by the functional food industry. And there are a lot of beneficial effects with fatigue, osteoporosis, obesity, and type 2 diabetes. And many of us are many of our food technology students and nutrition students and nutritionists and dietitians in the hospital, dietary planning and development of the foods, particularly the functional foods in various kinds of the diseases. We are incorporated these three different kinds of functional ingredients, pro, pre, and symbiotics. So there are a lot of applications in functional food products. If you see, in recent years, in the COVID-19 pandemic situations, the more people are preferring taking probiotics. So that increase in knowledge of functional foods in recent years and health benefits beyond the basic nutrition. So that functional foods not only giving basic nutrients like energy, protein, vitamin, and minerals, they are an additional health benefit they are providing. That's why they are becoming that much praise among the consumers. The presence of probiotics in commercial food products have claimed certain health benefits. This is very important. So this is this particular situation and scenario in recent years led to the industries focusing on different application of probiotics in food products and creating a new category of functional food products as probiotic health foods is one of the category that emerged in the functional food product class. So these are all the some probiotics in functional food product development. In these products are also include some prebiotic, and few of them are already symbiotic. So majority of them are from dairy-based probiotic foods because milk is one of the very important, very convenient ingredient. We can easily incorporate probiotics because it facilitates the growth and multiplication of probiotics. It is very good vehicle for the incorporation of either probiotic or prebiotic. So many of the dairy-based probiotic foods are available. Like yogurt is one of the very demanding product, and number of variations, number of different yogurts are available in the commercial market right now, and drinkable fresh milk and fermented milks, cheese, etc. So apart from these dairy-based probiotic foods, many non-dairy-based probiotic products are also available because it's the sake of vegans and vegetarians. So these probiotics are incorporating into various kinds of functional food products which are non-dairy based, and vegetable-based probiotic products are available. Vegetable fermented juice, carrot fermented juice, beetroot fermented juice that are there in Amazon health food section, and fruit-based probiotic foods, and cereal-based and meat-based probiotic. Meat-based probiotic foods are very traditional category. We know that the sausages and dry sausages, which are very famous fermented meat-based product. Again, meat industry is experimenting with different novel kind of sausages by incorporating various functional ingredients and make it as more functional. The functional sausages are available in the market. While we are developing functional foods with special reference to pro and symbiotics, there are a lot of application challenges that we have to face. Either the researcher or the food industry has to face many application challenges. Because you know that probiotics are live microorganisms. So when we are experimenting with live microorganisms, when we are feeding with live microorganisms to the consumer, particularly the patient, a lot of care must be taken while formulating, while designing the functional food products with either probiotic or symbiotic. So what are the challenges generally facing with regard to probiotics and symbiotics? The strain selection. So appropriate strain selection has to be done, and screening, because if we take one particular category, lactobacillus, if we take lactobacillus as a culture, started culture, there are various strains of lactobacillus are available. So which particular, actually what exactly that we want to prepare, for what purpose, and which lactobacillus species will perform that particular function in that product, and what are the interactions that happens between the ingredients, and is it really fit into the food matrix? There are various things that we have to consider and screen the strain, and then only we have to select. So this is the most challenging part of the development of functional foods with probiotics. And the other challenge is growth and survival during processing, and the viability. Viability of probiotics is very important and very difficult in product development. in functional food product development with probiotics. Without viability, there is no value for the probiotic product or symbiotic product. But protecting the viability, protecting the survival of friendly bacteria in the product is the most difficult task. If we have to produce in a large scale in the food industry to improve, to extend the shelf life of the product, so a lot of things are involved. So the storage temperatures and processing temperatures and using of sophisticated kind of processing equipment for the development of the product. And these are all the various things that, and packaging material, the use of packaging material. So these are all the various things that have an influence in the survival of probiotics as well as the viability of probiotics. So this is the most challenging task of the researchers and the technologists in functional food product development. And sensory properties, the effect of sensory properties. This particular probiotic, anyhow, when the organism is working in the system, working in the food, it releases certain kinds of odors, which are sometimes not acceptable. And the taste sometimes, the mouthfeel after consuming certain kind of prebiotic food. So these are all not acceptable in many of the sensory evaluation studies. So keeping the sensory attributes in a right condition is very, very intelligent task that has to be performed while, developing the functional food. So that's why we had to mask the flavor of it. We had to mask the mouthfeel of that particular off flavors and off taste with other ingredients. That's why, many functional food product development with probiotics always include a certain kind of flavor from herbs or a certain incorporation of fruit pulp or vegetable juice into it to make more appeal in terms of color and in terms of flavor as well as in terms of taste. So masking of that particular off flavors which are produced during the fermentation process of activity of these particular microorganisms is very, very important point that has to be keep in mind while working with probiotics in development of functional foods. And the other very important thing that I want to mention is the safety assessment. While working with probiotics, those are all living organisms. So a lot of care must be taken while working with probiotics and symbiotics. Because, live microorganisms in the probiotic food or any food while we are working with a living microorganism, so few percentage of organism are also harmful to the body. Certain kind of pathogenic organisms may develop while during processing of probiotic foods and symbiotic. So we must take a lot of care with regard to the safety of the consumer health. Because consumer, when we are labeling as probiotic food, symbiotic food, consumers are blindly taking and consuming that particular food. So safety of developing functional foods with pro and symbiotics are very, very important. We have to take care of the strain and species and genus of the microorganism safety while incorporating into the product. And I already told you that viability is very, very important. The viability and the activity during functionality is one of the things that we had to check before sending into the market. So after developing the prototype itself, the researcher and technologist has to check the viability and activity of the probiotics in the product. Otherwise, even in India, we are not facing that much of laws and regulations with regard to functional foods. But in Western countries, it is very strict. We had to prove the viability and we had to prove the activity of the probiotics in the prototype development itself. Then only it is allowed for the scale up of the product in large scale by the industries. Then only you have to mention in your labeling of probiotic foods. And the adherence to the intestinal epithelium is one of the very important points that we have to keep in mind. And the lastly and very, very strict regulation is incorporation of food incorporation of probiotic in food, that has to be tested with volunteers or the patients with clinical trials. Then only probiotic and symbiotics are allowed for commercialization in functional food market. And there are many health claims that has to be remembered in labeling of probiotic and symbiotics, because food labeling and following the labeling protocols are very important in the promotion of any food product development, with special reference to functional food product development. So without scientific evidence and facts, we do not mention the term probiotic or symbiotic in the label of our functional food. And many of the manufacturers, they are used as a gimmick for the promotion. So based on the scenario, based on the demand of the product, they are just using the probiotic, the term probiotic and symbiotic. They are using misuse of the terms that generally happen in the health food market. So that's why International Scientific Association for Probiotics and Prebiotics, they made some recommendations. So in those recommendations, the first recommendation is they changed the definition of probiotic, prebiotic, and symbiotic that I already told you while I am introducing the terms in previous slides. So we had to follow the recent definition according to the regulation. And the other one is it's now required to include the microorganism species in the probiotic label. It is very important. And even the prebiotic, the name of the prebiotic has to be given in the label, the food label of functional food that has to be marketed. Because, many of the probiotic and symbiotic, prebiotic foods are beneficial for intestinal problems, gut problems. And majority of the people are suffering from inflammation in the bowel, and with regard to the celiac disease and gastric ulcers, etc. And they are choosing probiotic and symbiotic as an alternative. So in these particular patients, again, gluten is one of the things that we have to restrict. We have to avoid in the diets of those particular patients. So that's why it is mandatory to give the name of the prebiotic in the label itself, so that the patient can see the claim of that, and then only they will buy and consume the food product. And specific claims like health claim, nutritional claim, functional claim, this has to be given in the label of functional foods. And the term probiotics are not applied for cultures associated with traditionally manufactured fermented foods with no benefit of health benefit, with no proven of health benefit. This is very strict. And the word probiotics can be used for the defined strains from human samples for adequate safety and efficacy evidence. So these are all the recent recommendations that are given by International Scientific Association of Pro and Prebiotics. So these are must while we are preparing the label for prebiotic foods in functional food category. Is there 5 minutes more for me to conclude?
Yes, ma'am, you can continue.
Yes, madam.
Thank you. And I just want to give very brief applications of our research with regard to probiotic and development of symbiotic foods in the department of in the Division of Technology, Department of Home Science, Tirupati University. And we choose obesity. Why we choose obesity is it's a leading lifestyle disorder. If you see in India, it's also one of the most common lifestyle disorder, irrespective of the age and gender. So that's why we choose obesity. And there is one more very important point that is there in obesity, because there are the strategies, the intervention strategies and food-based strategies towards mitigating weight gain or overweight or obesity. Generally, we are seeing the etiology of obesity. So there are a lot of factors are involved, including the genetics or other things. But in latest research findings with regard to overweight and obesity, the gut microbiota is responsible, is one of the important factor in obesity, because the gut microbiota, the composition and the diversity of gut microbiota and the way that is responding towards the digestion and absorption of the food and storage of the food is quite different from obese people when compared to the lean people. This is the finding that is observed in recent medical research. And this suggests that there is an important role is played by the gut microbiota in the development of obesity, where we are generally neglecting this particular part while developing any intervention strategies for many of the diseases. So this information may represent a major advance in obesity therapy, since modifying the gut microbiota through a diet rich in probiotics or symbiotics can become an important treatment option for obesity. So that's why we choose obesity as because it's a leading lifestyle disorder, and we developed a few symbiotic products in the department. So I just share with regard to one particular product, the design and development of functional yogurt. We developed a functional yogurt. This is a symbiotic yogurt. For the ingredients that we used for formulation of functional yogurt are skim milk, because we use skim milk because we are targeting overweight and obesity. And again, skim milk, we did a lot of minimal processing, because we had to reduce the fat content of the milk. Simultaneously, if we reduce the fat content of the milk, what happened? The texture of, we didn't get the proper texture to the yogurt. So this is again the major problem. If you use skim milk, if you use low fat milk, we didn't get proper texture of the yogurt. That's why we had to do some kind of minimal processing to the milk. So actually the skim milk, we procured the skim milk with 5% of fat and 12.5% of SNF. So that much SNF is not sufficient to get a proper textured yogurt. That's why we modified the composition of the skim milk and increased the SNF to 14.5%. So that is the ideal SNF percentage for proper texture of the yogurt. And the prebiotic that we used for symbiotic yogurt is psyllium husk. Actually, we experiment with various kinds of dietary fibers, but we didn't get the proper acceptability with other different fibers. So finally, the psyllium husk, because it's very soluble fiber, it easily incorporated and give the proper functionality and the proper sensory characteristics to the yogurt. So then we finalized picking psyllium husk, choosing psyllium husk for incorporation into the prebiotic yogurt. As well as psyllium husk is one of the very, very important fiber, and they are having a lot of weight reducing benefits. In some studies already use this psyllium husk for weight reduction. So this is also one of the reason why we choose psyllium husk for the development of functional yogurt. And the probiotic cultures, lactobacillus and it's a mixture of probiotic culture that we procured from National Dairy Institute, and we used gargum as stabilizer to get the proper texture to the yogurt. So apart from the yogurt, we also developed a certain kind of other probiotic and symbiotic products like drinking yogurt and other things. And this is the composition and minimal processing of the formulation. I'm unable to give much details with regard to my products, because we just applied for patent. We filed for the patent. We also conducted the clinical trials, because it is mandatory. Whenever we are working with the living organisms, we should conduct the clinical trials before applying for the patent and before approaching the industry for commercialization. So according to the recent regulations, we already conducted clinical trials by taking permission with the ethical committee, and we got very good results. But I'm sorry, I'm unable to share my results this time. and we applied for the patent recently. And there are a lot of scope and future perspectives with regard to the probiotic foods. The youngsters and the young researchers who are working on food product development, particularly functional food product development with probiotic, prebiotic, or if you are developing with symbiotic, you have to take a lot of care towards designing your product. Because we know that the viability of microorganism is very, very important. While we are producing in a large scale, probiotic food in a large scale at industrial level, to the survival of microorganism to that much temperatures and the storage temperatures is very, very difficult. That's why you just apply the sophisticated processing techniques. So in recent years, encapsulation technology as well as nanotechnology is applying for designing functional probiotic and symbiotic probiotics. So you can work out, but it is giving very good results. And the count of it and count of probiotics and the viability of probiotics is excellent. While if we applied encapsulation technology, even the nanotechnology, and we have to work towards, applying the novel technologies with minimal loss in the activity, functionality, and the diversity of probiotics in the product. This is one of the major challenge that we are facing while producing probiotic foods in a large scale. And this is the one that I want to just conclude with this particular quotation. So everybody, you know that let food be the medicine and medicine be the food. This is the quote of Hippocrates, 5000 BC. This is the quote that is given at 5000 BC. But in present scenario, we can change slightly and make it like let functional foods be the medicine. Thank you very much for giving me this opportunity. Thank you.
Thank you, ma'am. Nice presentation. You are given a lot of information regarding prebiotics, probiotics, and symbiotics. And you developed a nice product. Congratulations, madam. You applied for that product for the patenting, all these things. It's a very nice thing. Now I open the session for any queries or any doubts. Dear participants, if you have any doubts.
Yes, Sirisha, I have a question.
Yes, madam.
Are you hear me, Sirisha?
Yes, madam. Yes, madam.
Okay. Manjula, thank you for your very nice presentation. It is very understandable to all nutritionists as well as non-nutritionists also.
Thank you, ma'am.
Very good presentation. And I want to ask you, do you have any data regarding how the gut microflora affect on diabetes? Because all gut viable microflora concentrate on gut area, how it affect on pancreas to reduce the diabetes? Do you have any data?
Actually, I don't have any work with regard to diabetes, the role of microbiota on diabetes, but I have literature with me right now because I studied extensively when I am planning probiotic and symbiotic foods for obesity. Because both diabetes and obesity are metabolic disorders. So I studied extensively with regard to the literature of both the ones. Ma'am, definitely I'll share you the literature, but I do not have any experience with the importance of it on diabetes, ma'am. Thank you. I'll share the data with you, ma'am. I'll send the data with regard to the diabetes.
Okay. Another question is, you said short chain fatty acids. It reduces the hunger in the gut, gut microflora and the metabolism. Even I have seen in the literature, most of the saturated fatty acids are re-enter into the energy cycle. Once the short chain fatty acids are produced in the large intestine, they reabsorbed into energy cycle or TCS cycle, and where they participate in the energy cycle to release the 2 kilocalories energy. So how this once they are reabsorbed, how they are creating this that is a hunger prevention effect in the gut area.
Actually, there is another function that is brain and gut axis is one more function. They're sending some neurological signals to the brain from gut area, where it sends the signal by feeling not to urge for the food and suppressing the hunger, etc. So that the individual feel immediately like that and responding accordingly, ma'am. This is one of the thing that is happening while the short chain fatty acids are released after metabolism of the food, ma'am.
Okay. And another question, you said many commercial probiotic products are producing now. They are in the commercial market. So once probiotic products are developed and after developing, they are entering to the commercial market, they give definitely expiry date. So once expiry date is over, after the expiry date is over, if any person consumes, is there any chances of producing these viable bacteria, the toxins? Is there any chance of producing toxins by these viable bacteria after expiry date? Do we have any data?
No, not toxins. I do not know about the findings. I didn't study the findings with regard to the toxins that are released after expiry date. But other undesirable microorganisms and the pathogens may develop, may multiplied. So after expiring date. So fermentation is one of the excellent technology. At the same time, if there is a protein molecule or protein component that are there in the food, putrefaction also happens after expiry date. So then it will become not, it will not acceptable for the consumption. That may happen.
Okay. So
Manjula, I will add that because I will add some more point for your discussion. That is mainly whenever these are the milk-based products. So the yogurts that are being prepared using the skim milk or whatever the milk they are going to use, they will be using the probiotic cultures in the milk-based products. So milk-based products shelf life is very short, and if it is exceeding the manufacturing date, definitely it is going to yield to the spoilage of that products. So that may itself is not the consumable form. So that is why it has to be under refrigerated conditions. If it is exceeding that date, surely it is going to affect the it is going to lead to some other disease conditions.
Yes, ma'am. Yes, ma'am. Apart from
diarrhea, mostly the diarrhea or continuous diarrhea is being reported in this type of consumption of this type of products.
Yes, ma'am. Actually,
irritable bowel syndrome, and diarrhea is the major problems that are going to be.
Yes, there are some inactivated microorganisms that are also there in the product.
Yes.
So that can be multiplied after expiry date, which leads to the problems, particularly gastrointestinal problems.
Yeah. So, Dr. Manjula, my appreciations are with you for your nice presentation. I have a simple question here. Generally, the children are very much fond of the sausages and everything. So, after making fermentation is the process to make the sausages. No, so we'll store them at the refrigerated conditions or at the room temperatures also sometimes. Suppose if we reheat it, or suppose if we apply any cooking procedure for that, does it kill the affect the good bacteria? Does it affect the
actually, madam? So many of the processed foods with longer shelf life, the microorganisms are in non-viable form, inactivate form. So I think they are more stable during short time processing, reheating in microwave. They can survive, they can stable. But prolonged heating and exposure to high temperatures, maybe the count and availability of the microorganism, that I do not work on that, but they are stable at minimal processing, minimal heating with microwave. That is already proven.
Okay.
We can microwave the product.
Okay, thank you.
When all probiotic products are developing based on the milk and milk products, and what about vegan diet? Can you comment on vegan diet, Manjula?
Ma'am, vegan diet with regard to the incorporation of probiotic and symbiotic, that is the recent applications that is happening, ma'am, in food industry. But how, in what form they are incorporating and they are generating the microorganism in vegan food, that I didn't look onto that till now, ma'am. We had to check.
Okay.
Mama.
So any more questions?
Madam, one question is there in chat box, I think. Can combine the culture with nuts, milk like almond, coconut, etc., madam?
Madam, there is a question like, ma'am, can probiotic grow inside of symbiotic, that is probiotic plus prebiotic microbreads by consuming the prebiotic?
I didn't understand. Can probiotic grow inside of symbiotic microbes by consuming the prebiotic? Jahnavi, Bhargavi Reddy, will you please ask the question once again? I didn't understand your question.
Yes, madam.
What exactly that you want to know? Because probiotics, generally the probiotics will multiply and grow and increase in number when we incorporated prebiotics by developing of the symbiotic food. In any symbiotic food or food product, the number of probiotics are increased. That we also get the same kind of results while we are developing symbiotic yogurt and symbiotic drinkable yogurts. So we compared the probiotic yogurt as well as symbiotic yogurt. The probiotic activity and the viability, when compared both the products, the viability and compatibility functionality of probiotics are high in symbiotic product when compared to the probiotic product that we observe. Is it the answer that you need, Bhargavi Reddy? Is it okay? Definitely, symbiotic have more additional health benefits when compared to probiotic alone.
Madam, there is one more question. Can combat the culture with nuts, milk like almonds, coconut, etc., madam?
Yeah, if your composition, with any kind of vegetable milk, during fermentation process, you may develop probiotic bacteria in your product.
Thank you, ma'am. Thank you for your answering. You answered almost all the queries. Dear participants, if you have any doubt, please post in the chat box, or if you want to contact with madam, I will communicate the madam contact details, all these things. Thank you. Now I invite Bindu madam to conclude the session. Thank you.
Thank you, Sirisha. To conclude this session, I extend my heartfelt thanks to Dr. Manjula, who has wonderfully presented the different aspects of probiotics, prebiotics, and symbiotics, which are the components that enhance the human health by several mechanisms. Madam, as you have rightly said that all of us would love to be healthy and fit. And nowadays, a lot of lifestyle disorders are getting increasingly common across the age group, irrespective of age and gender, which can be attributed to various influencing factors, like it can be because of the lifestyle which we choose, or it can be because of our shift from the traditional to more processed food consumption, stress, lack of sleep, less physical activity, and also all of us are being adopted to the advanced technology which are within our reach. And you also has mentioned about the statement of Hippocrates, where all diseases begins in the gut. Yes, as you have rightly said that what we eat is what we are. So that is the reflection where we can perceive from an individual. Hence, all of us should mind what we eat, what we cook, and even what we prepare in a holistic act. So that is also a takeaway component which we have to understand. And one more quotation also you have mentioned, that is heal the gut and prevent the disease. Yes, nowadays in order to counteract with the lifestyle disorders, all of us should look upon our gut health. So that is also very much important. And Madam also has highlighted various importance of human gut microbiota and its interaction in development of various dietary strategies. Like she also mentioned about vital functions such as digestion by involving the metabolism, and also the vital biosynthesis, immune modulation, and also role of microbiota in stimulating hormones from neuroendocrine cells of gut in promoting satiety. So that is a novel concept where we need to look upon in order to treat the lifestyle disorders, Madam. And one more new concept you have addressed is that is regarding the occurrence of dysbiosis, which is also a new term for me, especially to understand this. And its causes and effects were also well explained, Madam. Thank you for elaborating on all these things. And one more thing is you have elaborated on therapeutic uses of foods, and also the application of foods and beverages, and also regarding the challenges in the development of probiotics and prebiotic foods, where it is a very much very essential and needed information regarding the various components to look upon while preparing a functional food. And you all, thank you, Madam, for this session, which is very useful and very informative to all of us. Thank you so much, Manjula, Madam. Over to you, Sirisha.
Yes, Madam.
Manjula?
Ma'am.
Congrats, Manjula. All the best. Good presentation.
Thank you, ma'am.
Once again, congratulations, Manju.
Thank you, Akka.
Nursing వాళ్ళకి కాలేజ్ పిలిచినారంట మా.
Before
Students have to come back to.
Manju, congratulations.
Ma'am, thank you very much.
Very good presentation.
Thank you, ma'am. Before concluding my session, I would like to thank all the organizers, Dr. Sirisha and Professor Arna ma'am, Professor Rajni ma'am, and Bindu, for giving me the opportunity, because this opportunity in presenting the seminar in Mahila University is very, very precious gift for me, because so giving presentation in a seminar conducted by my mother institute, it's really honor for me to give the presentation in the seminar, because I am one of the alumnus of the Department of Home Science, Mahila University, and giving my presentation in front of my teachers, that is very, very nice experience that I have today with Jyoti ma'am, with Taruna ma'am, and Vijay Jyoti ma'am, who are my teachers. And Rajini Akka, of course, not my teacher, but she is, I can say that she's a guide to me since my post-graduation, giving a lot of suggestions in improving myself in research and profession. And thank you, Shirisa, considering me and my work and giving opportunity to share my research in this seminar. Thank you very much, Sirisha and Bindu.
You are always welcome, ma'am.
Thank you.
Thank you, ma'am.
Thank you, ma'am.
Next speaker, Rekha Sushil Kumar madam is already joined. Now I invite Ambati Kimera introduce today's present session speaker, Rekha Sushil Kumar Madam. I invite you, Kimera.
Thank you, ma'am. Good afternoon to everyone. It's a great pleasure for me to welcome our next speaker for today, who is going to talk about functional foods and nutraceuticals. This is a subject in which we should be deeply interested, as it gives us the importance of foods which are highly nutritious and associated with powerful health benefits. Before going into the session, I would like to give a brief introduction about the speaker, Dr. Rekha Sushil Kumar Sharma ma'am, presently working as Associate Professor, UGC, Academic Staff College, Nagpur University. She is actively engaged in research and training. Her research interests are food science, nutrition, dietetics, clinical nutrition, higher education, and ICT. She took up her master's in food science and nutrition from Nagpur University, MPhil in food science and nutrition, Nagpur University, PhD on nutritional evaluation and storage studies of wheat germ. Also did her PG diploma in higher education from Indira Gandhi National Open University. Ma'am has started her professional career as honorary lecturer at postgraduate teaching department of home science, Nagpur University. Was a counselor in IGNO's DFSM programs at Rashtrasan Tukoji Maharaj Nagpur University and at Sadabai Raisoni Women's College. Held positions as research assistant in projects like nutrition, health education and environmental sanitation, mother-child healthcare, conducted by UNICEF Nagpur. Was lecturer in Institute of Hotel Management and Catering Technology, Vice Principal and Officiating Principal in Tulu College of Hotel Management, Nagpur. Also coordinator for MOOCs Swayam Functional Foods and Nutraceuticals. Ma'am has handled and is handling PhDs, where 4 are awarded, 2 submitted, and 6 are ongoing. Ma'am has guided M.Sc dissertations, 14 in food and nutrition, 3 in DFSM programs. She has published 1 book, Wheat Germ, a nutritional capsule, research papers in 4 international journals, 38 national journals, and 8 in reputed journals and periodicals. In conference proceedings, 1 international, 10 national, and 3 book chapters. Ma'am has presented in 17 conferences and was co-author in 28 conferences. She is a life member in 6 organizations: Nutritional Society of India, Indian Dietetic Association, Society for International Development, Rome, Italy, Geriatric Society of India. Held various positions in local bodies as Secretary, Joint Secretary, Executive Member, Board Member, Vice President, President in organizations like NSI, IDA, AFSTI, Geriatric Society of India. Ma'am has received 1 Young Scientist Award and 15 Best Paper Awards. Today, with immense pleasure, I welcome you, ma'am, to enlighten us on functional foods and nutraceuticals. Now the session is over to Dr. Rekha Sharma.
Thank you, Dr. Kimera. Can you hear me?
Yes.
Am I audible?
Yes, yes.
Yes, ma'am.
Yes.
I'm sharing my screen. Thank you for the elaborate my introduction. Please let me know whether my screen is visible or not.
Madam, it is visible, madam.
Okay. So thank you so much at the outset. I thank Shri Padmavati Mahila Vishwavidyalayam for organizing webinar on foods for human health. And I thank organizers, especially Dr. G. Sirisha, for giving me this opportunity to share my presentation on functional foods and nutraceuticals. In fact, I'm having cold and cough and other problems today, but I thought not to miss this opportunity, and I'm here to give my presentation. So, dear participants, I will be giving the overview of functional foods and nutraceuticals. Just now, I was listening to Dr. Manjula, very in-depth presentation of prebiotics, probiotics, and symbiotics she has given. But I'll be then just giving you an overview of this. I won't be going into the details, but just an overview. Let me start with my presentation. So, yes, before I start, let me tell you my course on functional foods and nutraceutical is available on SWAYAM portal. And this program, this is already launched on 10th of January 2022, and you can register before 28th February 2022. See, if you go to swayam.gov.in and if you write in course catalog functional foods and nutraceutical, you will find my course. And till date, about 835 students have enrolled. And let me also tell you, this is the 6th time my course is being launched through this SWAYAM portal, and about 10,000 students from 74 countries have joined till date. And students, I think, of food technology, food engineering, biotechnology, then M.Sc nutrition and dietetics, they all can join for credit transfer. Otherwise, dietitians, nutritionists, they can join this for updating their knowledge. So this is the detail of my course. This is like you can take down later on also. Last date of enrollment is 28th February 2022. There is no registration fees, but if you want to appear for the exam and want to get a certificate, you need to appear for the exam, and the examination fees is 1,000 rupees. But if you just want to update yourself related to functional foods and nutraceutical, you can join and you can attend the course. So today my presentation will deal with what are functional foods, what are nutraceuticals, what are bioactive compounds, and I'll be giving you few examples of functional foods and nutraceuticals. So let us start with what are functional foods. I think this is the second day and you must have learned a lot of information on functional foods. But just to revise once again, the Functional Food Institute USA in 2017 defines functional foods as natural or processed foods that contain biologically active compounds which in defined, effective, and non-toxic amounts provide a clinically proven and documented health benefit utilizing specific biomarkers for the prevention, management, or treatment of chronic disease or its symptoms. Then what are nutraceuticals? You know, these two are two different terms. So nutraceuticals, they are chemical components of food when ingested in appreciable amounts demonstrate attributes such as good bioavailability, bioefficacy, and exert positive effects on health characterized by disease prevention and alleviation. Now, let us see this also you must have seen in these 2 days, Dr. Stephen DeVillis, MD, coined the term nutraceutical, which is a hybrid or contraction of nutrition and pharmaceutical. And Japan is also the first country to have formulated a specific regulatory approval process for functional foods. In 1991, the concept of foods for specified health use, that is for sure, was established. And in US, the term nutraceutical products are regulated as drugs, food ingredients, and dietary supplements. Now let us see what is the difference between functional foods and nutraceuticals. So functional food is like, this is turmeric. Turmeric is known as functional food because it has a bioactive compound. We all know it is curcumin. So curcumin, when it is along with this food, it is known as functional food. Whereas nutraceutical is when this curcumin is isolated, extracted, and it is sold in the form of powder, pills, or in the form of tablets, then it is known as nutraceutical. So the basic difference between functional food and nutraceutical is functional food is the bioactive compound is inside the food in its own form, And when this is extracted and sold in the form of pills, powders, or in the form of tablets, then it is known as nutraceuticals. Now, let us now study what is bioactive compounds. Bioactive compounds are biologically active, which indicates that the substance has a positive effect on living organism, tissues, or cell. Now, they play an important role in human growth and development and have proven health benefits. They also amend disease risks by easing disease conditions. So what are these bioactive compounds that also we'll see? They are not nutrients. Like I think there are students of nutrition also. There are non-nutritionists also. We all know that carbohydrate, proteins, fats, vitamins, minerals, water, fiber, they are nutrients. But these bioactive compounds are not nutrients. They are not essential for life. Like nutrients, we all know that they are essential for our life, and if they are not present, deficiency diseases occur. But in case of these bioactive compounds, they are not essential for life, but likewise perform essential functions. Then these bioactive compounds have been defined by National Institute of Health as constituents in foods or dietary supplements other than those needed to meet basic human nutritional needs, which are responsible for changes in health status. Now, these bioactive compounds, they have health promoting effects, and to name a few, they act as antioxidant, anti-inflammatory, anti-diabetic, anti-cancer, anti-arthritis, prevents cardiovascular diseases, prevents neurological disorders. So we'll study this in detail later on in later slides. So let us now study some of the examples of functional foods and nutraceuticals. They are probiotic, postbiotics. Right now, the earlier speaker has elaborated this. Then prebiotics, symbiotics also she has elaborated. Then non-digestive carbohydrates, polyphenols, pigments, then organosulfur compounds, glucosinolates, saponins, hemagglutinins, and bioactive compounds and spices and condiments. So I will be covering few functional foods and nutraceuticals. So this she has elaborated just now. Morning. Hello? Is there any problem, Dr. Shirisha? Okay. So probiotics she has just now given. It is live microbial cultures of a single strain or mixture of different strains that beneficially affect the host animal either directly or indirectly by improving its intestinal microbial balance. Now we all know that lactobacilli and bifidobacteria, these are two species which are important or they work as probiotic. Lactobacillus, they are present in all milk and milk products, but bifidobacteria are not present in any foods. They are present in our large intestine. In our intestine, they are present, and it is like the growth of these bacteria. They depend upon the type of food we consume and the medicines. If we consume the antibiotic, so this will kill all our good bacteria inside our body also. And again, the basic difference between yogurt and curd. In a few places, yogurt and curd is considered the one and the same thing, but there is a difference. Difference is of the organism. Yogurt especially contains Lactobacillus bulgaricus and Streptococcus thermophilus, whereas our curd contains Lactobacillus acidophilus, Lactobacillus lactis, and other microorganisms. So that is the basic difference. Now, the very important thing is live microbial culture. That is after, like before serving of this food, the food should not be cooked if this is to be served, if live microorganism is to be served. I was listening to one of the question of the attendees that does microwave, this keeps the organism alive if the food is reheated in the microwave. Even I have not come across with any of such studies, but I think a slight temperature will kill these microorganisms. Then the another term is, these are all the mechanisms. I will not be going through this. But there is another term known as postbiotics. Postbiotics is if you're making idlis, dosas, dhokla, then kadhi, using curd and using fermented food, all the microorganisms, they get killed. But the bacterial products in the absence of viable organisms may have similar effects on signaling pathways and barrier function. They act similar to probiotics, even if the organism is not live, microorganism is not alive, but its byproduct will give the similar function inside the body. These are broadly characterized as postbiotics and can be defined as non-viable bacterial products or metabolic byproducts from probiotic microorganisms that have biologic activity in the host. And then, for example, bacteriocins, organic acids, ethanol, diacetyl, acetaldehydes, and hydrogen peroxide. So even if in the cooked fermented foods, microorganism is not alive, but they have similar activity as probiotics. So, and then in the last slide, I've given you an example of, you all must have seen in Maharashtra, it is very common to use to make Gopal Kala during Janmashtami. And it has a mixture of curd and all vegetables and cereals, millets. So it is one of the best mixture of probiotic and prebiotic food, where the food is not cooked after adding curds. So any food which is not cooked after adding curd and consumed, that is a very good source of probiotic. Then this also she has explained. So prebiotic is mostly fiber that are non-digestible food, but and beneficially affect the host's health by selectively stimulating the growth and or activity of some genera of microorganisms in the colon. I have been talking about bifidobacteria. So if we consume these foods, they are not digested in the intestine, but in the colon, they get fermented, and then they promote the growth of microorganisms. And these prebiotics are oligosaccharides, resistant starch, dietary fiber, and gums. Now let us study these one by one. Now before this, we will see right now, like I told you about probiotics, you have also studied what is prebiotics. Now a mixture of prebiotics and probiotics is known as symbiotics. So symbiotics are a combination of both pro and prebiotic. The combination is such that a particular live organism, the growth of organism is more in a presence of certain prebiotics. So symbiotics are often defined as synergistic mixtures of probiotics and prebiotics that beneficially affect the host by improving the survival and colonization of live beneficial microorganism in the gastrointestinal tract of the host. Then one by one, we will study these prebiotics. The first is oligosaccharides. We know that depending upon the number of saccharide groups, they are classified as monosaccharides, disaccharides, polysaccharides. Oligosaccharides means the saccharide or the carbohydrates which are made up of 3 to 10 saccharide units. And the functions are they enhance beneficial bacteria such as bifidobacterium. Yes, it is a very good source, or it promotes the growth of bifidobacterium species and lactobacillus species in the colon. Then it increases the digestion of lactose, enhance mineral absorption, improves the good HDL and low density lipoprotein, then decreases serum lipids and decreases blood cholesterol. It also improves blood pressure. So these and they decrease fecal pH also. Okay, so these are the examples of. There are several oligosaccharides: fructo-oligosaccharide, galacto-oligosaccharide, lactose, sucrose, malto-oligosaccharides. If you do my course on functional foods and nutraceutical, we have given a very good account of these oligosaccharides. Dr. Renuka Mainde has presented this oligosaccharide chapter. then soya oligosaccharides. And you know, the sources are like arvi, you must have heard about, soya bean, even raw onion, and this, they contain a very good amount of oligosaccharides. In our Indian tradition, we have a culture of consuming onion, raw onion along with the food. So that might be one of the reason why we include it in our diet, in our meals, I should say. Then comes resistant starch. Resistant starch is the portion of starch that escapes the digestive enzymes in the small intestine, which is resistant to digestion, passes further into colon. So this is we all know that starch, they are made up of amylose and amylopectin. And then amylase is needed for the digestion of this resistant starch, for the starch. But inside the body, some of the portion of starch cannot be digested. They get escaped in the digestive enzyme in the small intestine, and it is resistant to digestion. So resistant starch that reaches the large intestine can act as a substrate for microbial fermentation, the end products being hydrogen, carbon dioxide, methane, and short chain fatty acid. So all these foods, you know, they contain resistant starch. And especially if you boil potatoes, keep it in the refrigerator, and after 1 to 6 hours, if it is taken out and if it is consumed by thawing, you'll find that it contains more of resistant starch. Even the same thing with rice. Rice, if it is cooked, kept in the refrigerator, and if it is consumed after 12 to 24 hours, the content of resistant starch increases. Again, you must have heard about bagara bhat or ponitsa bhat. Ka matlab, like if the rice, leftover rice is kept in the refrigerator and consumed on the second day, it is good because it contains more of resistant starch. So there are 5 types of resistant starch. There is RS1, RS2, 3, 4, and 5. RS1 is, it is physically inaccessible starch and present in all coarsely ground or whole kernel grains. Then resistant starch to RS2 is present in high amylose maize starch, raw potato, raw banana starch. RS3 is the one which I was talking about, retrograded starch, which is present in cooked and cooled starchy foods. I think it is good for diabetics also, like if they want to consume rice, but if they consume this type of rice, the glycemic index of these product is very low. Then RS4 is chemically modified starch, which contains cross-linked starch and octanyl succinate starch. Resistant starch 5, RS5, contains amylose lipid complex. So this, there are different types of resistant starch. And yes, dietary fiber, all know about this very well. There are 2 types, insoluble and soluble fibers. And you must have learned about this in wide detail. But all these, like oligosaccharides, resistant starch, dietary fibers, and gums, these all 4 things have a common function, that they are not digested in the small intestine, and they are carried towards colon, where they are fermented. And after fermentation, they help in the growth of microorganism probiotics, which are present in the organisms, which are good for our health. Then the 4th is gum. You must have seen these types of laddus, which are usually given to lactating mothers. For especially, it contains good amount of calcium. It is good for health. But the important thing is, again, it is not digested in the intestine, and it is carried towards colon for fermentation. And especially it is gum arabic, which is said to be very used in our Indian dietaries. They are found in woody tissues of some plants, and they are secreted by specialized cells of the site of injury to form exudate, which immediately solidifies on exposure to the atmosphere. And they act as prebiotic. And then they have several therapeutic actions such as hypoglycemic, anti-diabetic, antioxidant, immunomodulatory, and anti-ulcer. There are various studies Like if 10 grams of gum is consumed for continuously 10 to 15 days, it leads to, like, it also affects the good obesity, like weight reduction also it helps, and it helps the good microflora of the gut microbiota also. Then polyphenols, yes, it is a large, it doesn't come under prebiotics now. They are again functional foods. Polyphenols are a group of chemical substances found in plants characterized by the presence of more than one phenolic group or building block or molecules. So polyphenol is
Madam, maybe some network issue, ma'am. Madam, you are in mute. Unmute yourself, please.
Am I audible now?
Yes, madam.
So we'll be studying flavonoids. Flavonoids, they are constituent of the largest group of plant polyphenols and are responsible for many of the plant colors with the brilliant shades of yellow, orange, and red. Then yes, tannins and catechins, they come under polyphenols. Tannins are water-soluble polyphenols that are present in many plants, fruits, beverages, vegetables, some grains, cocoa, chocolate, and beverages such as coffee, tea, and wine contains tannins. Then catechins, they are the most active constituent polyphenols in green tea. You must have seen advertisement in TV, like instead of doing a physical activity, you should consume tea that gives you that it is better than doing the exercise, as it is said. Then they are found in red wine, broad beans, black grapes, apricots, and strawberries. Then phytoestrogens. Phytoestrogens is a female sex hormone. Now, when the source is a plant, it is known as phytoestrogens. So dietary estrogens are naturally occurring compounds found in plant which have a structural similarity to the female sex hormone. And the examples are soybean and flax seeds. Then carotenoids. So carotenoids, as a student of nutrition and dietetics, you must be knowing about beta carotene and vitamin A activity. So these richly colored molecules are the sources of the yellow, orange, and red colors in many fruits and vegetables. Alpha carotene, beta cryptoxanthin, lutein, zeaxanthin, and lycopene are the most common dietary carotenoids. Now we'll see few carotenoids. Like first is beta carotene. Function is it can be made into vitamin A in the body. Then it neutralize free radicals that may damage cells. They support cellular antioxidant defenses. And the sources are yellow, orange fruits and vegetables like papaya, apricots, carrots, pumpkins, sweet potatoes, bell peppers. And alpha carotene is found in palm oil, maize, carrots, and pumpkin. Then lutein and zeaxanthin, they support maintenance of eye health and prevents cancer. And sources are green leafy vegetables like spinach, lettuce, broccoli, kiwi fruit, egg yolk, corn, and citrus fruits. Lycopene, yes, it decreases prostate cancer risk, prevents other cancers, reduces symptoms of exercise-induced asthma, prevents atherosclerosis. Let me tell you one point here. Now, instead of raw potato, if tomato sauce is consumed, that gives more better, or the bioavailability of lycopene is more when it is consumed in the form of sauces. Then food sources, most red fruits and vegetables like tomatoes, processed tomato products, watermelon, pink grapefruit, guava, they are good source of lycopene. Then curcumin, we all know about turmeric and curcumin and its benefit, and we know that it is a very good anti-inflammatory. I will be showing it in the later slides. Now anthocyanins, they are red to blue color pigments and present in purple grape, purple sweet potato, purple carrot, black bean, purple bean. And if you have ever heard about kokum, it is a very good source of anthocyanin pigment. Then, yes, organosulfur compound. Yes, in many houses, this garlic, yes, it is crushed in the form of chutney and it is consumed along with the meals. It is a very good habit of consuming such type of chutneys because, you know, while crushing this garlic, allicin, actually injury to the garlic bulb activates the enzyme allinase, which metabolizes allicin into allicin. Now, allicin is a very good bioactive compound that is good for health, and it is produced only if garlic is crushed and then it is consumed. Then glucosinolate, all these cruciferous vegetables that includes cabbage, then broccoli, then cauliflower, and these products, they contain glucosinolates, a group of sulfur-containing glycosides present in cruciferous vegetables said to be protective against cancers of lungs and alimentary tract. Then yes, we have now come to a very important, that is bioactive compounds in spices and condiments. We all know that India was a place for trade during when the Britishers were there, and the major reason was spices. So these spices and condiments, they are food adjuncts that are used for thousands of years to enhance the sensory quality of foods. The quantity and the variety of the spices consumed in tropical countries are particularly extensive. These impart characteristic flavor, aroma, and color to foods, stimulating appetite, as well as modify the texture of food. Recent research reveals that dietary spices in their minute quantities has an immense influence on human health. A wide range of beneficial effects on human health by the action of gastrointestinal, cardiovascular, respiratory, metabolic, reproductive, neural, and other systems. So a small, this thing I'll tell you, a reference. The cancer incidence in countries such as in India is 94 in 1 lakh. Okay. But in United States, it is 318 per 1 lakh people. So this suggests the potential role of spices in cancer prevention. Because we consume good spices in our diet, in our meals, that helps us to prevent from cancer also. Now I'll be showing you few spices that contains a bioactive compound. Like ginger contains 6-gingerol. We during cold and cough we consume this. Then quercetin, onion contains quercetin. Then clove contains eugenol. Then black seeds are also known as kalonji. It contains thymoquinone. You know, it is given in the literature that it can treat all types of diseases. It can treat all types of cancers, except that it can cure everything. That is how it is written. Then allicin, just now I have given you. Then piperin, black pepper contains piperin. Then red pepper contains capsaicin. Then cinnamon contains cinnamaldehyde. Then saffron contains crocin or quercetin. And fenugreek seeds contains diosgenin. Now, these are some of the examples, health benefits of spices like garlic, onion, fenugreek, turmeric, bell pepper. They lower blood cholesterol. Curcumin, capsaicin, they prevent and dissolution of gallstones. Then curcumin, capsaicin, garlic, they protect erythrocytes integrity in hypercholesterolemic condition. Then fenugreek, garlic, onion, turmeric, cumin, they are all, they have hypoglycemic potential. Curcumin, onion, amelioration of diabetic nephropathy. Then turmeric, curcumin, capsaicin, and eugenol, they have antioxidant effect. Then turmeric, curcumin, capsaicin, eugenol, they have anti-inflammatory and anti-arthritic effect. Then turmeric, curcumin, garlic, ginger, gingerol, mustard, they have anti-mutagenic effect, and that is cancer preventing effect. Then again, curcumin, capsaicin, piperine, ginger, cumin, ajwain, fennel, coriander, onion, mint, they have digestive stimulant action. I think because of this only we use black masala, garam masala in our sabzis. Then turmeric, curcumin, garlic, asafoetida, they are all antimicrobial. Then, yes, I have made a table of all the spices with their bioactive compounds, the name of active bioactive compounds. Like I've shown you, ginger contains 6-gingerol, rosemary contains rosmarinic acid, onion quercetin, turmeric curcumin, clove eugenol, fennel flower thymoquinone, black pepper piperine, garlic allicin, red pepper capsaicin, parsley apigenin, oregano or celery contains luteolin, capers contains camphorol. You know, if you attend our course on functional food and nutraceutical, you'll study all these bioactive compounds also. So then again, saffron, quercetin, crocin, fenugreek, clove contains eugenol, cinnamon contains cinnamaldehyde. Then basil, yes, basil and cloves both contains eugenol, you know. That's why again during cough, what we do, we make kadha. You must have heard about Ayush kadha also. So what they use basically, clove, basil, cinnamon, they all contain this eugenol in a very good amount. And then fennel, coriander, sesame seeds, basil also contains uracilic acid. Then mint contains carbone, nutmeg contains myristicin. Then here I have presented the name of different bioactive compounds along with the activity, like they have antioxidant activity, then anti-inflammatory activity, anti-diabetic activity, anti-cancer, cardiovascular diseases, they prevent cardiovascular diseases, and they also help in the treatment of neurodegenerative diseases. Yes, this is about all spices and condiments. These are all the bioactive compounds present in spices and condiments, and these are all the activity they do. Here you can see that for neurodegenerative diseases, resveratrol, quercetin, camphorol, piperine, then rosmarinic acid, apigenin, thymoquinone, they play an important role. Then yes, we'll now come down to omega-3 and omega-6 fatty acid. Since I have made this presentation for a lay person, like anybody who doesn't know about what is this omega-3 and 6 fatty acid, I've given the details of the structure of fats. So Dr. Shirisha, I would like to know whether I should go through the basics or should directly jump over to the details. Okay.
Continue, ma'am.
Okay. So chemical composition of fats. Fats, they are made up of one molecule of glycerol and 3 fatty acids. Hence, then it is known as triglyceride. So one molecule of fat, glycerol with 3 fatty acids. Okay. Now, what is fatty acid? Now, this fatty acid, each fatty acid has a an omega end or known as methyl end, or which has CH3, 3 hydrogen and 1 carbon, where another end is a acid group or has a COOH group. This is one fatty acid. Okay. Now, fatty acid consists of chains of carbon atoms with a methyl group of CH3 at one end and carboxyl group at the other end. Now, the general formula of fatty acids is this, we will not go again. Now, second is these fatty acids are classified according to the degree of saturation. If there are no double bonds in the carbon chain, it is known as saturated. If there are double bonds in the carbon chain, it is known as unsaturated. Okay, so saturated fatty acids have single bonds between carbon atoms, and unsaturated fatty acids have one or more double bonds between the carbon atoms. Now, let us know what is omega-3 fatty acid and omega-6 fatty acids. So, from the methyl or the omega end, if we count the number of carbon atom having the double bond, first double bond, that is known as, from this end when we count, so first, second, third, fourth, fifth, and sixth. At sixth level, sixth carbon atom, there is a double bond. So this is known as omega-6 fatty acid, which is linoleic acid. Okay, and this is first, second, and third. At third carbon atom, there is a double bond. There is the first double bond. There are 3 double bonds in the structure, and the first double bond is at third carbon atom. So this is known as omega-3 fatty acid. And this is an example of alpha-linolenic acid. These are known as polyunsaturated fatty acid because they have double bonds in their structure. Okay, I think the concept of this omega-3 and omega-6 fatty acid is now clear. Now in the fatty acid, from the omega end or the methyl end, the first double bond in the carbon chain in this fatty acid chain, when we see, that is according to the number of carbon atom, it is known as omega-6 or omega-3 fatty acid. Now, we'll also see the concept of cis and trans fatty acid. You must have heard about TFA, that you should consume fat which does not have trans fatty acid, or the amount of trans fatty acid less. So, cis isomers are those when both hydrogen atoms are placed on same side of the double bond. When these hydrogen atoms are placed on the same side of double bond, it is known as cis. And when in trans isomer, the hydrogen atoms are placed on either side of the double bond, this is known as trans fat molecule. So omega-3 fatty acid, it is an essential fatty acid as it cannot be synthesized in the body and must be supplemented in the diet. Then 3 types of alpha-linoleic acid, eicosapentaenoic acid, EPA, and docosahexaenoic acid. They are omega-3 fatty acids, and AL is precursor of long-chain fatty acids, eicosapentaenoic acid, docosahexaenoic acid, which have many health benefits. Again, you must have seen in the advertisements also, ye khana chahiye because it contains DHA. Okay, so omega-3 fatty acid sources are flax seeds, chia seeds, and walnuts, cod liver oil, fish oil, canola, soybean are also sources. Omega-3 fatty acids involved in the several mechanisms of atherosclerosis, thrombogenesis, and stroke. Then during pregnancy and breastfeeding, it is crucial in development of central nervous system of infants. That is important with DHA. Then yes, omega-6 fatty acid. This is very important. You know, we have never heard about the grass eating animals. You know, those cattle which are fed on this grass, they contain the milk of such animals. They contain more of omega-6 fatty acids. So the meat and milk of grass-fed ruminants offers the richest sources of natural conjugated linoleic acid or omega-6 fatty acids. So examples are whole milk, cream, butter, cheese, and whole milk yogurt from grass-fed animals. They are said to be excellent sources. Yes, I have kept few slides for antioxidant also, because I think many students, they do not have idea of what is antioxidant. They talk about this, that the particular food is very good antioxidant, but what is basically antioxidant is, here I've given you. Yes. So antioxidant.
Detailed about the antioxidants, ma'am.
Yes.
We heard about only the word.
Yes, yes. So antioxidants, they protect the human body from oxidation brought about by free radicals, superoxide and other. This we will see now. So this is a stable molecule. When any item, it has the outermost layer, if it has all paired electrons, this is said to be a stable molecule. Okay. But if a free radical comes, free radical means a atom which have unpaired electron, that is it does not have a pair with it. So what it will do, it will start snatching an electron from the stable molecule, and while doing it, it disturbs the whole metabolism and the other things related with that stable molecule. It will snatch the electron from a stable molecule. So if an antioxidant is present, antioxidant has large number of electrons in its outermost layer, which can give it to the free radical. If this electron is given to the free radical, it gets satisfied and it does not affect the stable molecule. It stops snatching the molecule from the stable molecules. So what we do if we have antioxidants, that is, we have fruits and vegetables where the outermost layer of yes, they give this extra electrons to such free radicals and they satisfy them. If these radicals, if they have unpaired electron, they will start creating millets and will not allow, or it will affect the cellular structures, DNA, and the mutation will occur and the other things will occur. So the best thing is to consume good amount of antioxidant. This will give electron to these free radicals. Then yes, how these free radicals, they can and the oxidative stress is given here. Now you must have heard about ORAC score, oxygen radical absorbency capacity, that how much antioxidant property they have. You know, it has all majority of fruits and vegetables, they are not Indians, so I'm not going through it, but you know, cauliflower, spinach, onion, it is given. I'm so sorry. Then yes, cinnamon ground contains good amount of antioxidant, ORAC units. Then turmeric also, and please remember, turmeric alone is not so effective, but it is effective more when it is given along with the black pepper. So mixture of turmeric and black pepper is an excellent medicine for all. So the functional foods represent one of the most intensively investigated and widely promoted areas in the food and nutrition sciences today. However, it must be emphasized that these foods and ingredients are not magic bullets or panaceas for poor health habits. Balanced diet is only one aspect of a comprehensive approach to good health. So thank you so much. I thank once again Dr. Shirisha and Padmavati Vishwavidyalaya for inviting me for this lecture.
Thank you, ma'am. And now the session is open for queries. You clearly explained about what is the difference between the functional foods and nutraceuticals, and which food contains the main component of this nutraceutical. All these things very clearly, madam. A very nice presentation. Thank you.
Thank you.
You are suffering with severe cold and cough again.
I wrote you in the morning, but then I thought you were timetable will be disturbed and how you will manage. And then I thought of doing it. I thought I was not audible properly, but then I tried my best, Dr. Shirisha.
It's very nice presentation, madam. Thank you.
Thank you so much.
Yes, ma'am. Now session is open for question. If you have any doubts,
I would suggest the participants to please enroll for the course. That will give them the good picture, because I cannot include everything. I cannot explain them everything about these functional foods. But if they join our course, they'll be like it will be very easy for them.
through your course, we identified you, ma'am.
Okay. Yes.
Our students are enrolled.
Acha. And you know, from all over India, it is your university. From maximum students, they enroll and they do it for credit transfer also. So I have mentioned many times to UGC also. These people have problem because their exams and markets. You get problem because of Corona, you know, the dates.
Yes, madam. Yes.
Yes, postponed. I think there are no questions. Either my lecture was very simple or maybe it was very difficult to understand.
No, madam, it's very useful, I think. All are enjoyed.
Thank you so much.
Thank you, ma'am.
No questions.
Very good, informative, ma'am.
Thank you.
Nice information you have shared with us.
Thank you so much, Dr. Aruna.
Dr. Aruna, ma'am, she is our HOD, madam.
Okay. Yes. Namaskar, madam.
Namaste. My camera is not working.
Yes. Dr. Manjula, madam, is from SB University, madam.
I'm very nice to meet you. I'm Dr. Manjula, Associate Professor.
I
know you. Yeah.
I was listening to your lecture because I was waiting for my turn, and I listened to it. It was an excellent lecture on probiotics.
Thank you, ma'am.
You can start adding probiotic, postbiotics also in your lecture now.
Yeah. Actually, the concept that I heard from your course itself, but I didn't work much on that, ma'am.
Okay.
Definitely, I'll take. Thank you.
But it's a good concept, you know, those who cannot eat raw, curd, because it is considered cold food and then it causes many problems. So these people, they can't consume curds, the kadhi and other cooked foods they can consume. Yes.
Dr. Bindu, madam, from our department, madam.
Okay.
Madam, please.
I couldn't see her. Namaskar to everyone, those who have invited me. Thank you so much.
Madam, namaste.
Namaste.
Namaste, madam.
Professor, retired Professor Dean from our department, Jyoti ma'am.
I've seen your photographs in the brochure.
Yes, Jyoti ma'am. And Dr. Madhavi, faculty from our department. Dr. Niharika.
Good afternoon, ma'am.
Good afternoon.
Dr. Beulah.
Nice to meet you all.
Yes.
Though we are online, but it is very, I'm very happy to see you all here and to meet you.
Yes,
hope you all have seen my course.
Once the pandemic is over, we will invite you, ma'am, to our department.
Yes. Sure.
You can come to
Please,
our university as well as
for the virtual photo. Yes. Dear faculty, all of you join for the virtual photo, please.
And Dr. Manjula, you're from Home Science, Food and Nutrition.
Yes, ma'am. Food Science and Nutrition from the Department of Home Science, ma'am.
Okay.
From Tirupati itself.
Venkateshwara University.
I belongs to Venkateshwara University.
Venkateshwara University. Lot of work you have done. Very good work.
Thank you, ma'am.
On probiotics, yes. Even if we are from this, we have to study a lot to know all these things, While doing this functional food course, I studied a lot, I did a lot. Our whole team, in fact, we did a lot to understand this because, when I was a student, I did my M.Sc in 1985 from Nagpur University. We had just one part as functional foods. I never studied this, but while making it, I referred all journals and books and what not to make this course. So it was not like available in any of the books. I used to refer all journals, LCHS journals and all good journals for making my like lecture notes and other things.
In 2002 itself, in CFTRI, they were working on this probiotics.
Yes,
2002. From 2000, yes. They were working on that. Because we went there for UNESCO.
To invite eminent personalities from our university for the valedictory function. They are Professor A. Jyoti Madam and Professor P. Uma Maheshwari Devi Madam. Professor A. Jyoti Madam was a former Dean of Sciences and Professor in our Home Science Department, who enmarked for her sincerity, passion in teaching, and excellent administrative ability. I would like to extend my valedictory invitation to Professor P. Uma Maheshwari Devi Madam, Head of Microbiology Department, whom we are observing her untiring efforts in her guidance for NAAC accreditation. I appreciate and congratulate all the team members of the conference for well planning and organizing the conference. Now I am fortunate to welcome our beloved Professor A. Jyoti Madam to address the gathering to this valedictory function. Good afternoon, ma'am. Please, you proceed with your valedictory.
Thank you, ma'am. Thank you, Bharati madam. Now I welcome Bindu madam to introduce our today's guest, Jyoti ma'am.
Jyoti garu, namaste. Namaste, ma. Chala రోజులు అయింది మా చూసి. Yeah, I miss you a lot. జ్యోతి గారు అని పిలిచి అలవాటు అయిపోయి మిమ్మల్ని పిలవక చాలా రోజులు అయిపోయింది.
Thank you, Sirisha. Good afternoon, one and all.
Sirisha ma'am, it is showing host disabled participant screen sharing, ma'am.
Yes, just a minute. Yes, now you can share. Bindu madam.
Thank you, Sirisha. Am I audible?
Yes, madam.
Okay. Good afternoon, one and all. I am very happy and delighted to introduce our professor, Dr. A. Jyoti Madam, Senior Professor in the Department of Home Science and former Dean, School of Sciences, Sri Padmavati Mahila Vishwavidyalayam, Tirupati. Madam has 35 years of experience in the department as a professor. And regarding her academic qualifications, Madam has obtained her master's degree from Acharya Nagarjuna University and PhD program from SNDT Mumbai, SNDT Women's University Mumbai. And Madam's areas of research interests are dietary management during disease conditions like renal diseases, diabetes, cardiac, obesity, anemia, arthritis, cancer, HIV, AIDS, etc. And Madam has already developed many nutritious products to combat deficiency diseases and nutritious related chronic disease, degenerative diseases. And regarding her publications, Madam has published 40 papers in national and international journals, and she had one book published and 5 book chapters to her credit. And Madam has, under her guidance, Madam has completed 12 PhD students and 2 MPhil candidates. And Madam has headed 3 major research projects. One is entitled as Anemia in Fluoritic Pregnant Mothers, Results Low Birth Weight Babies, Rectifying Through Elimination of Fluoride and Control Through Nutritive Diet. And the second one is Madam has also conducted DST inspired programs in the campus twice. And Madam also headed a very prestigious Rural Women Technology Park as a principal investigator. Regarding the awards, Madam has obtained Best Paper Presentation in IDA program in 2009 and 2013. And Madam also has organized and conducted various many exhibitions on important days, like on World Food Day, Diabetic Day, Renal Day, Science Day, Nutrition Week, HIV/AIDS Day, and even Consumers Day. With this brief introduction, I would like to welcome our beloved Jyoti Madam to this valedictory function. Thank you, Madam, and welcome you, Madam.
Thank you, Bindu, regarding nice words about me, and our Bharati for inviting me as the valedictory addressee, and Sirisha, convener of the seminar, today's seminar, and the university authority for inviting me as a valedictory addressee, and other faculty members of our department, Home Science Department, head Aruna, Rajni, contract faculty, and then students and other today's seminars beneficiaries and other members. Thanks a lot for inviting me. So I would like to talk upon I headed that is I was a principal investigator for the Rural Women Technology Park, which is funded by the DST seed. It's a major research project. 3 years we have conducted and completed in 2020. So where we developed so many products. So that's why I would like to present all those things before you. And if your people are interested, we can transfer the or disseminate the knowledge as well as the technology to you people to start up your own enterprises also. So actually the final year students who are interested can come forward and take the technologies from the department. You can even use the equipment from the department, which is handed over to the department. You can use and develop products, and you can sell it also. And we have also taken a FASI license. So even that also can be utilized by you under the name of Spurti, which is given to the products, and take the administrator's permission and pay the minimum charges or something, if whatever they fix, and you can start up your own enterprises with the help of all these things. So today I'm going to present the nutraceuticals developed already since 2 days. We are talking about the nutraceuticals, functional foods and other things. And my previous speakers has given you what is the difference between functional foods, nutraceuticals, and what are the functional foods, how they helps the or gives the benefits to the health of the people, and nutraceuticals, how to develop and what are the different functionalities in the foods, and then how it has to be safely used, etc., etc. I think you are well versed now with all those things, so I need not tell about different things. But we have to recollect the 2500 years back, Hippocrates has given food will be the medicine, and make the food as your medicine as your food or something. So that phrase is so much essential nowadays, because it is having since 2500 years back it was there, and they have developed, invented many things, and also many functionalities in foods, and how they react, how it helps us, everything we have seen. And now it is more the important, and everybody is aware of the things. Food is having so many of benefits. It is the must to or mandatory to have to control many of the conditions, especially the COVID-19, which is a pandemic condition, which is most worsening the condition. If only with the help of the foods, etc., we can develop our immunity, and we can be free from that COVID-19, etc., etc. So everybody is now clear about that. Food gives the rectification from the so many of the disease condition, even the pandemic condition COVID-19. So that's why we have to be learned many things, how to prepare, how to utilize, etc., and free from all this one. And especially we have to boost our immunity, then we are free from many conditions. Even a simple element that we think that iron is the one with the deficiency of iron, we say that if there is a deficiency or low hemoglobins, nothing will happen. But that is the only condition which will lead to many of the chronic conditions, like a heart attacks, then diabetes, whatnot, each and every chronic conditions. So that's why we should not, as a students, you should not, youngsters, you should not neglect the iron as well as other nutrients like calcium, magnesium, all those things which are so much helpful for the people, along with the major nutrients like carbohydrates, proteins, fats, etc., etc. So vitamins, minerals, everything is essential nowadays. During a pandemic condition, we have to more concentrate on the vitamin D. It is also essential to control many of the chronic conditions. So the foods which we developed are having many of all these keeping the view of all these things, and we developed many of the products. Around 30 products are developed, and these products can be stored for a minimum of 6 months, and it can be utilized for a long run. And it can, so that's why the people who are interested to become as entrepreneurs, please take up the technologies and start up your own enterprise, and make the people healthy and prosperous. Okay, so the functional foods used in preparing these nutraceutical products from the RWTP are herbal products like tulasi, tender mango leaves, even that is what is that, different varieties of fruits, vegetables, etc. And grains we use, green leafy vegetables we use, spices we use, and utilizing all these things and its functionalities, we developed around 30 products. So you know that tulasi is the one which is very well grown and easy to harvest, easy to cultivate. And yesterday Rajni has asked Madam, what is that, when should we harvest tulasi or something. The tulasi is a not a seasonal plant. It can be grown anywhere, any place where we have a little bit of a space, like even a pot can be used to grow that. And it can be harvested in the around 4 to 5 p.m. and you can have its whole benefits. And tender as well as the matured leaves also have the same type of benefits, etc. And it is more helpful for each and every ailment, like a simple thing, cough, cold, to higher conditions like controlling the sugars, body fat, etc., etc. So we have developed a products using this tulasi leaves. And we have tulsi patti powder, that is where we used only tulsi. Since it has a controlling anemia, it can boost the bone health. Not only that, it helps in many of the conditions. So we used separately a tulsi patti powder. And then we have prepared a rice roti mix also, the wheat roti mix by using this tulsi patti powder. And toffees we prepared with ginger, without ginger also. And then tulsi fryams we developed, and they are liked by many people. And tulsi toffees with the cloves also. So since they have all other ingredients in it, like herbs and spices, etc., they have so many of benefits like anti-aging properties, antibacterial, anti-spasmodic, and immune boosting, laxatives, anemic, anti-anemic, etc., etc. So tulsi can be used. And as a students, you can also develop many products out of this tulsi, since it has so many of the health benefits. And next to that tulsi, we have used tender mango leaves, especially this red mango leaves, that is the very tender mango leaves. They have the anthocyanins, and they are much benefits in it. We developed using this mango leaves, the spicy mix we developed. And we prepared a combination of ginger and other things. We prepared a toffees, herbal tea powder, and by using ginger flavor, the biscuits we have prepared, and then roti mix also we prepared. So keeping all these things, like it has so many of benefits, like boost the digestion, anti-hypertensive, and anti-diabetic, anti-laxative, and anti-ulcer effects, spasmodic, anti-spasmodic, anti-diarrheal, detoxifying, mild laxative, and antibacterial, etc., etc. This tender mango leaves can be used. And already we know that matured mango leaves we use as a torna, and it gives the oxygen to us and helps to eliminate the infections entering into the home, etc., etc. Even tender mango leaves can also be used, and they are more benefits. Even simply if we chew the tender mango leaves, that is the red colored, are more beneficial for the diabetic and other things. We have studied many of any variety of the mango leaves also has the same benefits. So you can use all those things. And since we have the products developed should stay for a longer period, we have used the dehydration technique by using the solar dehydrator, where we can minimize the losses of nutrients during preservation, during drying process, etc. So we have prepared a ginger garlic powder. So it is so much useful for the day-to-day cookery. Ginger garlic is used for many food preparations. So that's why we can use this ginger garlic powder. And also we kept it in mind that is the more of the people are going nowadays to foreign countries, where they get a difficult to get the ginger garlic, so they can take this ginger garlic powder. So internationally also it can be sold. That's why we have developed this ginger garlic powder. It is especially enhances the immunity of the person, so it can be used, and also improves the white blood cells. And even singly also we can prepare the ginger separately, garlic separately, or combination of ginger garlic also. Ginger is the one which is helpful from the simple element like the common cold, cough to the even the obesity and other things. Nowadays, if you look into the YouTube channels for reduction of body weight, especially they uses this ginger in the for the preparation of the decoctions, etc., etc. So ginger is most powerful functional food which we can be used to prepare any of the nutraceutical products. Next to the ginger, we have developed a lemon powder also with the help of a lyophilizer and they have what is that spray dryer, and pineapple powder also we prepared, which has so many of benefits like reduces the inflammatory properties. And pineapple is one thing which the bromine which is containing in this is helpful for reducing the body weight. So it can be used for many of the many of the health conditions, anti-aging properties, and lemon is also can be used in many things like weight reduction as well as immunity, giving immunity, and reduces inflammatory actions, etc. And even it has the anti-aging property also. So fat reduction, cancer controlling, etc. So this lemon powder can be used for everything. And we have planned to develop a tablet-like things, and which can be used for easily dissolve in water, and it can be prepared into lemon juice, fresh lemon juice like thing, etc., etc. Next to this, we developed amla powder. And amla, you know that it not only gives the immunity to the people, and also it's because of its antioxidant property, it not only anti-inflammatory, it has a reduces the discoloration of the hair also. So in henna powder or in preparing any of the decoctions for the hair growth and hair coloration, we can use this amla powder. Even if you eat amla powder, it gives you so much of a immune boosting, etc., etc. Next to sapota powder, the our IFT student Haripriya has developed this sapota powder, and it can be used like a milkshakes. It can be so, and since of its functional properties, it can be prepared into reconstituted into a milkshakes, or otherwise it can be used in bread, cakes, etc., etc. It can be used in any form and to get its benefits, original functional benefits. Next to it, we developed a virgin coconut oil. This is why we call it as a virgin coconut oil, is they developed from the fresh coconut, and this is we can use it in the we can develop it in two ways, dry and fresh, but fresh is more beneficial. It consists more of a lauric and caprylic acid, myristic acid, etc. And we seen that we did many studies, and studies says even it controls the HIV/AIDS also. So that's why it can be used, and it is so costly also. It is it can be we get more financial benefits out of this preparing a virgin coconut oil. It is so easy, but we have to take so much of a precautions not to infect with other things, contaminate with other things, or otherwise it has so many of health benefits, like it controls the AIDS, hypertension, diabetes, and other things also. It even as during COVID also, if you have smear to the nostrils, the entry of the what is that virus is minimum and reduces the condition, COVID occurrence, etc. Next, tomato powder. You know that why we use tomatoes in different products, because it enhances the taste, as well as it consists of so many of the functionalities like flavonoids and antioxidants, free radical activity. It reduces the free radical activity. Even vitamins, many of vitamins are there, and which helps not only giving a coloration to the product and taste to the product, even it helps in many of the gives many beneficials also. It increases the RBC count, etc. It improves the blood condition, purifies the blood, etc. So that's why also we can use this tomato powder. So during lean seasons, it can be used even in many products to improve its taste as well as health benefits. So keeping that in view, we have developed a tomato powder also. Next. And then moringa leaves powder, that is drumstick leaves powder and wheatgrass powder we developed. And you know that moringa is nowadays, if you anybody have any foreign friends, etc., you just ask what is the thing. Nowadays, the people are more back about the moringa leaves, moringa powder, etc., etc., because they have seen it is giving many of the health benefits, right? Controlling the diabetes, hypertension, and even obesity, and increases the fiber, where we can control many of the chronic conditions, etc. And it is high in vitamins, minerals, especially the iron, etc. So people are more back about this moringa powder. So those who wants to have a international trade can develop moringa leaves powder. And wheatgrass powder is also, it can be easily grown at a household level also, and it has so much of vitamins, minerals, etc. And it has a more of health benefits. It helps in neurological disorders, as well as since it has so much of a fiber in it, it helps in many of the controls many of the chronic conditions also. And it is a it can be easily grown and prepared. Even it can be used fresh, but if it is not possible or otherwise to have an international trade, etc., you can develop and sell these things. Wheatgrass powder, easy and colorful, attractive, and much of the health benefits in it. Next. So like this, we developed many products, like 30 products were developed, and those things were with the help of this seed money, the DST seed money. We have trained around 340 rural women and semi-urban women, and also amongst them we have even trained the final year students of that batch, 2019-20 batch. And we have given training in all these 30, transferred the technologies to them. And those are these few pictures. You can see the action, and many seminars conducted on that. And we have transferred the technologies to that. And we have also, during that period, 3 years of period, we have conducted many of exhibition come sales, and some of the products were sold very well. Even it is the exhibitions conducted on campus and off campus also, and many people like those products and given a very good feedback to this. And like hotcakes, they were sold, and we made money from that. And technologies transferred to all these people. And even now, if people are interested, we can give the technologies to the people who are interested to become entrepreneurs. But take the authorities permission, and what is the modalities for that, etc. And I think I mentioned already to you that the Spurti is the name given to the product, and we have taken a license for FASI also. That is, Spurti is that Sri first letters of the Sri Padmavati University Rural Technology Enterprise. So keep with the help of that total thing, we have made given a brand name to the products as Spurti. Under Spurti name, you can develop and sell all these products, since we have a FASI license. So people who are interested, please contact the Aruna, or otherwise Jeevana Jyoti from pharmacy, since the Dean Nagaraj Garu, and you can ask them to transfer or take the modalities to develop and sell those products. And this is the thing, if you use all these products, you can simply take the functionalities of out of all these things, and you can help the people to boost their immunity and be healthy and develop a healthy nation. Thanks a lot for giving me this opportunity. Once again, I congratulate Sirisha and Sujata, conveners and co-conveners of the 2 days seminar, international seminar, and then head for giving me this opportunity, and other members of the committee, and beneficiaries who have listened patiently my talk. Thanks a lot. And finally, I thank the Niharika for presenting my slides. Since I am at home, I'm unable to share the PPT. I requested Niharika to. So thanks a lot. Thanks a lot for giving me this opportunity. Thank you.
Thank you, madam. Thank you for your active participation in our valedictory function. Thank you so much. Now I introduce next valedictory speaker, Professor Uma Madam. Beulah, please present the slide. I'm happy to introduce our Professor P. Uma Maheshwari Devi Madam and IQAC coordinator. Madam mainly working in microbiology department, and he received her postdoctoral fellowship at the Department of Molecular Technology, Penn State University, USA, and almost Madam completed 13 projects. In this, 8 is completed and 5 projects are ongoing, and almost more than 90 papers published in all reputed journals. Madam published books and chapters almost more than 12. Madam received international fellowships, mainly Boys Cast Fellow and Crest Fellow from the Department of Science and Technology, DST, and Department of Biotechnology of Government of India. And Madam received so many national fellowships like UGC Mid-Career Award from the UGC. And these are some other awards, mainly in the 2017, Madam received a State Best Teacher Award, and in 2021, Madam received AP Scientist Award. And mainly Madam area of research is, her focus is on natural products and therapeutic applications, with a long-term goal to explore and exploit marine microbiota for cancer and diabetes, Madam developed a diagnostic kit for identification of Anthogen's papaya disease. So the kit is very useful for the plant breeder to screen resistant variants from susceptible variants. In addition, Madam developed nanoparticles for the degradation of plastic and removal of textile dye colors from the water. Madam actively participating in almost in our university activities. Now I welcome you, ma'am.
Thank you, Sirisha, for the nice introduction. So many, you elaborated my biodata. First of all, I extend my sincere thanks to the Aruna, head of the department, and the convener, G. Sirisha, and R. Sujata, the co-convener, and my friend, Dr. Rajani, for giving me the opportunity. Maybe I think that I'm out of box of this nutraceuticals as well as the functional foods. But when I'm discussing with Sirisha, when she asked me that, definitely I can present a paper. But I can take you to the different world because this is maybe useful for the student friends who are there in the platform. So I thought that I can contribute something to the maybe the food microbiology. So you relish the nutraceuticals, maybe the nutritional foods and the functional foods. You tasted all the foods. Jyoti Garu had taken you a lot of herbal products for through her valedictory address. But I can take you, maybe I can give you a glimpse of that fear about the foods also. When you are taking the foods, you have to observe about, you have to think about the microbiologist before tasting the foods. Let me share my PPT so that I can take you to the technology world where exactly you can go for, you can look into that, the impact of the microbiology or the microorganisms. I think the slides are visible to you.
Yes, madam.
Yeah, let me take you to the, what are the molecular methods which are useful for detection of the pathogens which are prevailing in the food system, which we can call it as the foodborne diseases itself. You know that we are relishing that is the fast foods, the traditional foods, and so many foods. But before taking the foods, maybe the just now you heard that the fresh fruits, the cut, maybe the leafy vegetables which are available in the malls, that is the commercial market also. Before taking that, keep an eye on them, whether they are really suitable for us. You have to think about any foodborne diseases are there or foodborne illnesses are there. What are the root causes or the sources of the foodborne diseases or the illnesses? Maybe through ingestion of water, or maybe the wide variety of the food materials which are contaminated with the pathogenic organisms, like everybody knows about the bacteria, and now totally we are terrified with the viruses, the parasites and fungi. These are very important and which can be easily lodged on the foods itself. So currently there is a widespread of global public health problem is nothing but the foodborne diseases. What could be the problems or what could be the symptoms? You can look about the fever, maybe the nausea and vomiting, dehydration, and stomach upset. Usually it starts with the stomach upset, and you can see the body cramps, and finally the major illness is about the diarrhea itself. So in addition to these pathogenic bacteria, some of the root causes or some of the sources is maybe you can say that the toxins and the chemicals which are also responsible for causing the foodborne infections. So what are the major causes of the foodborne illness? So if you categorize them, the 66% of the foodborne diseases are mainly due to our friends, that is the bacteria, and some part that is the 26% is the chemicals, the 4% is from the viruses, and 4% is from the parasites. What are the predominant bacteria we can encounter during the foods? That is easily the bacteria, they can dwell on the food, on the surfaces of the food. You can see that the major bacteria which you can observe in the foods is, I think majority of the food technology students are here. My focus is on the students. You can learn in a better manner how to go ahead with the interdisciplinary, that you can opt for the food microbiology also for your research. This is my concern here. So that the bacterial counterparts like the Salmonella typhi, you know, the typhoid fever, Shigella toxins are there, that is a dysentery occurs, and the E. coli, which is the diarrhea, that is you can see about the role of the E. coli also. In addition to these bacteria, even the two important heroines of the foodborne diseases are, one is the norovirus, another one is the hepatitis virus. These are, we can consider these are the major contributor for the foodborne diseases here. When we are discussing about the foodborne diseases, always we can classify them into two types. One is the foodborne infections. That is, the infection is caused by the ingestion of the food which contains the pathogenic organism. Just by the ingestion of the food, that is the pathogenic organisms, through the food, the pathogenic organisms enter into the body, then they cause the infection. We can call that infection as the foodborne infection. The second one is, it is not the pathogen, the toxins which are produced by the pathogens, which are predominant, which are freely available in the food, that causes that ingestion of the toxins through the food, they can cause another disease which is called the food intoxication. So basically, it may be either due to the ingestion of the pathogens through the food, or maybe due to the consumption of the toxins through the food. These two are responsible for causing the foodborne diseases itself. Okay. So here, the integrated approach, that is both the qualitative detection as well as the quantitative detection, it is a very important in order to ensure the safety of the foods throughout the food supply chain. Because the food supply chain, whether it is a nutraceuticals or the nutritional things, or maybe the functional foods, the packaging or the food supply chain is very important. You have to be very, very careful from the first step to the last step, how it is ensuring, how it is free from the microorganisms, how it is free from the chemicals or the bacteria or the viruses. So that you have to be very careful about inspecting or detecting the foods while going through that the laborious process of the food supply chain itself. So my intention is to tell you about what are the different methods which are available in order to identify the pathogens which are there either in the form of the pathogen or in the form of the toxics itself. So you know that basically whenever we are speaking, the methods means the conventional method or the traditional method, and another method is called the rapid detection method. The conventional method, majority of the students, they know about this method. So whenever it is the food sample preparation is there, then our target is to detect the pathogens. We can go for that, how the pathogens are there on the surface of the food or within the food can be identified by culture techniques. Normally, we can prepare the agar plates, and finally, we can, by using the biochemical methods, we can identify that. It is very inexpensive. Every laboratory can practice this conventional method. Only the problem is here, it will take a lot of time. So the detection of the pathogens, we have to take the different media, the culture media, then enrichment media. Then if you want to know about the specific pathogen, selective media, then you have to isolate, and finally you have to confirm by the biological test. So this entire process, it will take, that is nearly 3 days to 7 days. So in the meanwhile, what will happen to the patient? So that they may be, that is, even the drugs cannot be prescribed without knowing that what could be the probable pathogen which is there in the food itself. During the, that is, everybody is facing this type of problem by having the gobi Manchuria, because this is against your functional foods or the nutritional foods. But anyhow, because of our tendencies or the lifestyles, always we can go for the fast foods itself. So that it is very difficult to give the treatment without knowing the, that is what could be the probable pathogen. Sometimes what will happen means the non-cultivable microorganisms which cannot grow on the agar medium. So if you are failed to identify that type of foodborne pathogens, it increases the transmission risk of the pathogens itself. When it is transmitted, what will happen for like a cholera? I know that the dysentery is transmitted in a very fast manner. So that the detection is very, very important in order to prescribe the drugs also. So in this scenario, there are several methods are introduced which are called the rapid detection methods. So here, instead of going for the culture method, we can adopt the culture independent methods. In the culture independent methods, directly we can take the sample, we can centrifuge the sample, and we can filtrate it, and we can go for that is the detection either by using the nucleic acid based methods. You know about the proteins, the carbohydrates, lipids, other counterpart is nothing but the nucleic acid, that is the DNA. So here we can go for the DNA or RNA based approach, or we can go for the immunological based methods or the biosynthesis. Out of these three methods, nowadays these nucleic acid based methods are very, very popular. So we can adopt this one, and the time which is taken for the detection of the foodborne pathogens is less than 24 hours. That easy for the doctors, easy for the patients. So immediately they can get the prescription and they can start the treatment also. So that the rapid detection methods are very important, especially for the identification of pathogens itself. So what are the molecular methods? That is the nucleic acid based methods. So these are the methods which are popularly used here. One is everybody knows that the polymerase chain reaction. If you can ask that is a 6th class student or the 2nd class student, they can easily tell about that. What could be the PCR? That is the thanks to the COVID. The COVID introduced the PCR into the common people also. That is a small, simple polymerase chain reaction. Then we can go for the multiplexing. Then you know about the real-time PCR, RT-PCR. Everybody knows about the RT-PCR. Then the nucleic acid sequence-based amplifications. Then the LAMP assays, that is the Luke-mediated isothermal amplifications. The ultimate method for the detection of the foodborne pathogens is the microarray technology. Slowly, I'm taking you to the high throughput technology, that is the microarray technology. I don't take much time because I want to tell you the basic objective of the molecular methods, how best you can identify the pathogens here. The simple polymerase chain reactions are very simple. That is basically here, that is pathogens can be identified based on their DNA. So that any single bacterial pathogen, it can be identified by using its DNA. How to identify? The DNA can be amplified by using the specific primers which are suitable. Primer means which is having the complementarity with the DNA molecule. So that with the simple method, with simple polymerase chain reaction, this is also called thermocycling reaction also. Why? Because it will run at different temperatures. So the simple PCR method is, we can take imagine this is the target sequence which is derived from the Salmonella, or maybe it is derived from Escherichia coli. These are the popular foodborne pathogens, that is the Listeria or the Campylobacter or the Salmonella or the Shigella, whatever it may be. So here, if you want to check about that, take the food, centrifuge the food and collect the DNA from that one, and you can run the polymerase chain reaction. It is performed in 3 steps. One is nothing but the DNA, you know that the double helical structure. The double helical structure is unwinded and each will act as a template. And in order to know about that, what we can do is we can add the primer and later we can synthesize that is what exactly called the DNA here. So based on whether the pathogen is having that is DNA, if the pathogen is having the DNA, easily it can be identified by means of the polymerase chain reaction. So for this purpose, we can amplify the DNA from the food sample, and finally we can identify what could be the nature of the organism. Is it the Escherichia coli? Is it the Staphylococcus? Or it is a Salmonella, which can be easily identified by means of the polymerase chain reaction. This is a very, very simple technique. So the same technique we can adopt. That is, I told you that this polymerase chain reaction is useful for identification of a single pathogens in the food sample. But we are not happy with the single pathogen. Sometimes the food may be contaminated with more than one pathogen. Under that cases, somewhat advanced technique is called, which is called the multiplex PCR. Everybody knows about the multiplex theater. That is, multiplex theater means the several screens are there. You can look about several cinemas within a mall. You can see the 4 or 5 screens are available. That type of things are called the multiplex theaters. The same principle was applied here. It is the multiplex PCR. We are running the same PCR, but this is meant for the identification of 4 or 5 or the 6 or the 7 pathogens at a time here. How to go for that? Similar to that of the polymerase chain reaction, here also we can take the different primer sets. So if it is the primer set is useful for identification of the E. coli, here for example, if it is having the combination of the 4 different bacteria, one is called the Salmonella, Staphylococcus, Shigella, and Escherichia coli. We can collect that is the primers for all. We can add it. If E. coli is there, it will express. If the Shigella is there, it will express. So that finally you can identify the availability of the genes here. Depending on the presence of the gene, we can say that whether the food is contaminated with only one pathogen or it is contaminated with the dual pathogens or triple pathogens, or maybe more than 3 pathogens can be identified just within a sample, single sample, and here the PCR PET which occurs at the multiplex manner here. This is a very popular technique in order to identify more pathogens in the food sample which are infecting which are contaminating also. Now I'll come to other technique, the real-time PCR. Everybody knows about the real-time PCR for the last 3 years, that is from 2020 onwards. We are running after real-time PCR. Everybody is going for the RT-PCR and they're testing and they're checking about the CT value, everything. So the real-time PCR, it works on the same principle of simple polymerase chain reaction. Simple polymerase chain reaction is nothing but amplification of the DNA molecule. But you may question me here. The COVID virus, coronavirus is, it is nothing but RNA virus, but you are saying that it's DNA. So here in the real time, what we are doing is we are collecting the RNA of the coronavirus. It is converted into the complementary DNA by enzyme, which is called the reverse transcriptase. And that can be the complementary DNA is used for quantification here. Just we are amplifying the DNA. In addition to the amplifying in previous cases, the simple and multiplex, what we did is, that is we identified pathogens depending on the amplification of the DNA. But here it is a real-time absolute quantification depending on the CT value. What is the CT value? That is nothing but a threshold cycle. Threshold cycle where exactly you can see the increase in the amplification process, rise in the DNA amplification, which is directly proportional to the fluorescent. In the other 2 cases, we didn't use any fluorescent markers where exactly you can go for the gel electrophoresis in order to identify the pathogens. But here it's a real-time absolute, directly you can measure that how much DNA is amplified, how much virus load is present. That's why we're taking the swab sample and going to that real-time PCR and they're analyzing and they're telling that what could be the CT value. Depending on the CT value, they're giving that whether it is a heavy load of the virus or the low load of the virus or the medium load of the virus, everything can be detected here. How it is possible means, here whenever we conducted the real-time PCR, the fluorescent probes are connected. That is the amount of DNA is directly proportional to that of the fluorescent probes here. So by measuring the fluorescent intensity, we can tell about that what could be the quantity of the DNA. What could be the quantity of either the viral pathogens or the bacterial pathogens which are available in the food sample here. Let me compare the 3 PCR methods. The simple PCR method, here single pathogen can be identified. Multiplex PCR, you can identify 6 to 7 pathogens in a single sample, whereas a real-time PCR, even the same genus with the different serotypes, the same genus which is having not only the species, I am telling about the serotype. E. coli itself is having 29 serotypes which are very, very impact on the food itself. That 29 serotypes can be identified by using the real-time PCR. So nowadays, majority of them, this is the threshold value. You can see that the quantity of the DNA, how the quantity of the DNA can be measured by means of the fluorescent intensity that is in the form of the CT value here. So that the multiple pathogens and the serotypes can be detected by using a important technique, the molecular method, which is called the real-time PCR. Now I'll take you another beautiful technique, which is capturing the entire global market, that is called the oligonucleotide DNA microarray. So this is awesome method. You can apply for the detection of any sort of pathogen which is present, even very, very minute quantity of the pathogen. Sometimes what will happen means one drug is given, the drug is not working. Even though the drug is given, it is not working because it is having the multiple pathogens, but one pathogen is at the lower level which cannot be traced by the several methods. That even lower minute concentration of the pathogen can be detected by the microarray technology here. So this microarray technology here, we can use the array techniques. Array techniques means maybe you can use the glass plate, or you can use the cantilevers, or nowadays we are going for high method like the plasmon resonant arrays here. Like this, these are the arrays. It's like simple glass plate which was graded. You can see that this is the glass plate which was graded. The blocks were created on the glass plate itself. So these glass plates, they're loaded with the probe. Okay, what is the probe? that is, this is a nucleotide fragment which is able to detect the other DNA molecule. Okay. So the probe is having the complementary sequence that of the DNA. You know that the DNA is a double helical structure, so that when the DNA is a hybrid is formed means when both are complementary in nature. Similarly, the same concept was used here. Here we can make the probe here. The probe must be complementary to that of the target. So it can be easily identified what could be the pathogen. My interest is to know about the E. coli, whether it is the diarrheal infection is there or not. So what I can do is I can make the probe which is suitable to identify the E. coli pathogen. Then I can take the sample here, the food sample here, which is contaminated with the E. coli. Maybe I can take the ice cream, or maybe I can take the beef sample, or maybe I can take the milk sample, or maybe I can take, you know that the fresh fruits are always contaminated with the salmonella. So that even the spinach also, I'm not terrifying you, but this is the fact. The spinach leaves which are available, sometimes they're contaminated with the salmonella also. So that I can take the sample from that. I can collect the DNA by means of the centrifugation process. Then here I'm taking the two samples, the sample A and the sample B. Then here I'm taking the mRNA from that. You can go for that. That is, mRNA is constructed. Then finally, I'm treating with that fluorescent agents. So the control and the another one is the target can be easily identified, especially by using the different colors here. That is, the different fluorescent colors are used. And finally here, it is probed with the top of the arrays here. These are the spotted arrays. When the single-stranded DNA is added to this one, then automatically what will happen is the probing occurs. After probing, because of the fluorescent dyes, you can see the emission. Whenever the light is emitted, when fluorescent probe is ligated with or complementary ligated with the DNA molecule, then automatically you can see that. Based on this, that is the dots and the colors, finally we can make the data analysis, and from the data analysis, we can conclude that what could be the pathogen and what could be the concentration of the pathogen. Sometimes it needs the antibiotic is combinatorial antibiotics are needed in order to treat the foodborne pathogens itself. So before prescribing that, the important thing is we need to know about what could be the foodborne pathogen. But as a nutritionist, as a home science student, what you learn is always be cautious about what are the possible ways of that is what the possible ways and the possible sources of food contamination. So these are the some methods. I know that some methods are I didn't concentrate it much because the time given to me is 10 minutes. I think, Sirisha, I don't consume much time. Thank you very much, Sirisha, once again. Thank you.
Thank you, madam. You are really taken into a different world. Always we are discussing about product and development process, testing, all these things. This is another angle of the testing of the quality of the product, all these things. Thank you so much. Almost you covered the detection of foodborne pathogens using different methods. Thank you so much for your presentation, ma'am. Now I invite Aruna ma'am to announce the best paper presentations.
One second, Sirisha, 1 second.
Madam.
This is Jyoti.
Madam.
The RWTP project is a multidisciplinary project. We have members from the pharmacy department, Professor Bharti Garu, Sridevi.
Your voice is not audible, madam.
And we, Vijay Lakshmi are the members. So we all together worked and prepared, developed, and shelf life studies and all other things are done with the help of all these members, and also the technology transferred, IICT training, etc. given by the communication and journalism people. So all these members are there. They are well versed with the products and other things. So you can approach anybody of the from the project, and you can get the information from that.
Thank you, madam. In our
Thank
in
you.
one meeting, present VC madam also discussed about this RWTP project, madam. Why you are not further taking?
Yeah, that's why I'm telling you better give this information to the outgoing students. They can become into an entrepreneur. So they can use these technologies. You can use the equipment present in the Department of Home Science. So even after completing their studies also, they can come and do their work, and so scientifically they can prepare the products and they can sell it. So modalities can be fixed with the help of university authorities, and make many entrepreneurs, food entrepreneurs. Okay.
Sure, madam.
That too, functional and nutraceutical products. Preparing a functional and nutraceutical. Even we have a tablet making machine in the Department of Pharmacy is there. Capsule preparation equipment. So all those things can be used. Some things we can given it to the this thing also. What is that? Central laboratory facilities.
Curie.
So all those things can be used and can become into an entrepreneur. So motivate the students to use all those things and become into an entrepreneur. Okay.
Jyoti garu, actually the pandemics made a lot of gap in the RWTP activities. Maybe Aruna can resume the activities with the support from other home science people. And other people like me and Usha definitely will contribute to RWTP whenever it is required. We are ready to, and me also. We are ready to contribute our services to the RWTP project. Whatever you want, definitely we are here to support you.
And I would like to mention one more thing for food fortification. I did an MPhil. My student has developed a food biofortified products. Okay, biofortified foods like using spirulina for as a menu. She developed many of grown many of the green leafy vegetables. All those things increased the iron, protein contents, etc., etc. So instead of going for all these processed products, even fresh things also can be helpful for the people. Day to day, they can even at home also they can grown up at the household level, like kitchen gardening or otherwise the roof gardening nowadays. Many people are interested about roof gardening. So all those things can be prepared by fortifying, biofortification. They can enhance their nutrient qualities and can be utilized day to day. Okay, so that also can be. If anybody is interested, you can contact me. I will tell you how to, etc. Thank you. Thanks a lot.
Okay, thank you, Sirisha. I would like to say a few words about the conference, and respected Jyoti Madam, former Professor and Dean School of Social Sciences, and Dr. Uma Maheshwari, IQAC Coordinator, and the Organizing Secretary, Dr. G. Sirisha, of this conference, especially and also my dear colleagues and dear participants. It is indeed a great pleasure for me to say a few words about this conference, and everyone has enlightened for these 2 days the important nutraceuticals and functional foods that are helpful in our diet, and also how it is going to prevent the disease conditions. So almost 30% of the population already are suffering from hypertension, and every 8th adult is urban Indian adult is suffering with diabetes, and they have impaired glucose tolerance, and also they are experiencing the childhood obesity, which is on rise nowadays. So there is evidence to suggest that moving healthy lifestyle is the only way to go out of these conditions, by consumption of healthier foods like functional foods, and also the nutraceuticals or the dietary supplements, and also the food portion, which has to be control over the food portion, and regular exercise. All these things will prevent and reverse some of the above disease conditions. So research and clinical evidences also have certain base mentioning that the use of these nutraceuticals and functional foods are helping a lot to overcome certain disease conditions. As Jyoti Madam has mentioned, that natural food products that are usually consumable products that are being used for isolating this type of nutraceutical, and that is functional foods that are helpful in preventing the disease conditions. So the speakers of today's conference have also mentioned, gave a the keynote addressee, Dr. Srinivas Janaswamy. He has mentioned about the bioactive compounds, which are sustainable tool, which can be used as sustainable tool for developing novel functional foods. So he has made a very good prominent research of his, mentioning that starch is also going to help in changing their structural pattern, may also help to maintain the glucose levels. So that is Dr. Naveen K. Rasdogi's chief scientist also mentioned, and he highlighted one of the new high pressure technology with a limited time is going to retain the nutrients that are present in the foods. And he also mentioned that the natural color has not been changed and sensory attributes are better in various foods with this type of technology when they have used it and tested in the laboratories. And one such company is also selling these type of products. And Dr. Hola Jo, and postdoctoral research fellow from Massachusetts University, USA, mentioned that fortification of plant-based milk protein, milk with calcium, fortification of plant-based milk with calcium may reduce the vitamin D bioaccessibility in vitro digestion studies, which she has carried out. And she has mentioned, she has taken the almond milk as an example, and she has mentioned her research related to that, and which is also giving an insight to the students that a new type of research and ideas can be generated into the students. And the other speakers like Dr. Pushpa Bharati, retired professor, spoke about millets in human health, and Dr. Manjula, associate professor from SV University, she has mentioned about, she has highlighted the importance of human gut microbiota and its interaction and its development of dietary which is a development of dietary strategies with regard to functional foods and nutraceuticals. And her main talk is on prebiotics, probiotics, and symbiotics as functional foods to address the lifestyle disorders, whatever is occurring nowadays. And finally, Dr. Rekha Sharma again gave a clear view about functional foods and nutraceuticals. So with all these type of speakers, and also the major presenters are the main people who have presented their research topics with enthusiastically, they have participated and they have shared their various scientific data and mentioned that these functional foods and nutraceuticals are essentially important in controlling disease conditions. And they have also mentioned that some of the cancers also have been prevented by using such type of bioactive compounds. So with this, it is come successfully, the conference has been come to an end, and I congratulate Dr. Sirisha, that is organizing secretary, who has taken pains. Without our help also, she could able to do a very good conference along with her teammates, Madhavi, Dr. Niharika, and Dr. Beulah, and Dr. Bindu, and Dr. Rajni. So I congratulate all of you.
It's a teamwork, madam. With all your help, I did this.
So with that, with their great planning and also the successful conduct of this conference, I congratulate all of you. And before leaving my platform, I would like to say that there are certain papers that are selected for best paper presentations, and this is on the basis of their time limit, how they have maintained their time, and also they're followed by based on their presentation skills, and also based on their papers that are being on the scientific papers. So I will announce now the best papers that are being selected. First paper is Muhammad Salem Elfark and Dr. Srinivas Janaswamy. They have worked on release studies of curcumin encapsulated in iota carrageenan cyclodextrin fibers during simulated digestion conditions. That is the best paper. And ICP 32, Aishwarya V. Patil and Pushpa Bharati. Development and efficacy of high fiber health mix from partitioned kodo millet. And ICP 4, Murthy Vidyarani, Galla Rajita, Umakant Nayak, and Aminin Uma Maheshwari. In silico molecular docking studies of selected nutraceutical against COVID-19. And ICP 17, Sindhu P and PB and Kulakarni UN. Buckwheat semolina, cooking quality and acceptability. I hope everybody is noting down the best papers. And ICP 30, Lata Rani R and Kulakarni, buckwheat upma formulation for nutrition security. K. Harita, P. Mahalakshmi, D. Sujata. In silico prediction of selected phytochemicals from Phyllanthus niruri for anti-inflammatory activity. Another paper is ICP 41, N. Haripriya, CHVSS, Kirti, Dr. M. Niharika. Development and standardization of purple yam edible waffles. And ICP 55, Shahana Mubin, Aruna, effect of supplementation of thermolact yogurt on serum calcium levels of 40 to 60 years age group. And ICP 58, Ranadeep Gogai, Sanjay Banerjee, Vinod Kumar Devarakonda, Rajita Galla, and Charumati Salva. Targeting on the evaluation of anti-cancer activity of proteins, peptides, using seed and seed coat of NIP 19. Then ICP 61, Vijeta Priyadarshini and Professor Gayatri Biswas, a study on food consumption patterns and nutritional status of tribal adolescent girls of Pionjor district, Odisha. And ICP 101, standardization, M. Kalavati and D. Madhavi, standardization of vegan yogurt from coconut and soya bean milk enriched with watermelon juice. So I congratulate all the best paper presenters, and your certificate will be sent to you by mail. Thank you. Thank you, Sirisha. And I thank I thank Professor Jyoti ma'am for extending her lecture and also giving a very good, beautiful insight with related to the powders, whatever she has developed, and she has worked very hard for that Rural Women Technology Park. And almost every day we used to get that entrepreneurs also to our department to see these products, whatever she has developed. I thank you, ma'am, once again.
Thank you, Aruna. Thank you.
Thank you, ma'am. Good afternoon to you all. Just I want to present a brief report on this 2 days international conference. Mainly, almost madam briefed about the speakers, what we discussed in the sessions, all these things. So here I am presenting a brief report on 2 days international virtual conference on nutraceuticals and functional foods for human health. So mainly the conference was organized in the virtual mode on 27th and 28th January 2022. The delegates from all over the country and out of the country have participated in this international conference. Almost around 350 number of participants attended the conference, and a total of 181 we received in this papers, almost some co-authors, all these things are there. 101 research papers and review papers were presented effectively in the 2 days sessions. And from this, we selected almost 11 best research papers were selected and announced. A total of 8 guest speakers from different eminent organizations.
Deepika, mute yourself.
Yes, different eminent speakers from different organizations acted as speakers. All sessions are very informative and useful. The inauguration session is started. Our Professor D. Mamata Madam, Registrar of the University, and Professor R. Nagaraju Sir, Dean, School of Sciences, inaugurated the inaugural session. Dr. Srinivasa Janaswamy Sir from South Dakota State University, USA, delivered the keynote address, and followed by Naveen Kumar Rastogi Sir from CFTRI, and Dr. Hulu Jo from University of Massachusetts Amherst, and Dr. Pushpa Bharati Madam from Dharwad University, and Manjula Madam from SV University, and Rekha Sushil Sharma Madam from Nagapur, and Professor Jyoti Madam from our parent university, and Professor Uma Maheshwari Madam from our parent university. These are all eminent speakers, delivered excellent information related to the conference theme. So I want to thank all these speakers for their acceptance. All deliberations goes on very smoothly. We are proud to announce the conference was conducted very smoothly. Now I invite Dr. Sujata to propose the vote of thanks.
Thank you, Sirisha, for giving me this opportunity. We have reached to the flag end of the 2-day international virtual webinar that has been successfully conducted. So on behalf of the organizing team, I'm privileged to propose a formal vote of thanks. Our program was successful only because of the support given by the administration. So our heartfelt thanks to our beloved Vice Chancellor, Professor Duvuru Jamunagaru. We like to thank our beloved Registrar, Professor D.M. Mamata Garu, for taking time out of her busy schedule to inaugurate this session, and also thank Professor R. Nagaraju Garu for his gracious presence in the inauguration session. This 2 days webinar will not be a quite success without the eminent speakers. So we want to register our heartfelt thanks, sincere thanks for all the speakers, eminent speakers who delivered the scientific deliberations for the 2 days. The sessions begin with Dr. Srinivasa Janaswamy. I know this is a repetition, but it's our duty to extend our thanks to each and every eminent speaker. So this program was successful because of Dr. S. Janaswamy from South Dakota State University for delivering the keynote address in the inauguration session. That was followed by the scientific deliberations by Dr. Naveen Kumar Rastogi, Chief Scientific Officer, CFTRI, Mysore. We thank you for your talk, sir. I want to thank Dr. Hula Ju, postdoctoral research from University of Massachusetts, Amherst, for sharing her scientific work. Despite of the time difference, she was able to make it and she was able to share her knowledge with us. Thank you, Dr. Hula Ju. We are very much happy and we are very much, I should say, blessed to have Dr. Pushpa Bharati Madam, retired professor Dharwad. She has been with us since inauguration. And I think I just saw her in the lecture session also. Thank you so much for your patience and your support for successful conduction of this event, madam. Dr. Manjula Kola, Associate Professor from SP University. We thank Dr. Manjula Madam for her scientific deliberations in this conference. And last but not the least, the person behind the Food and Nutraceuticals MOOCs program, that is Professor Rekha Sushil Kumar Sharma, associate professor from Nagpur University. She has delivered a beautiful talk on functional foods and their functions. Thank you, madam, for your presence. This 2-day session might not have been possible without the team. So I should register thanks in front of all the August gathering to the team. The team consisted of Head of the Department, Home Science, Professor M. Aruna Garu, Dr. N. Rajini Garu, Dr. V. Bindu Garu, Dr. D. Madhavi, Ms. A. M. Beulah, Dr. M. Niharika, and I cannot miss the convener, Dr. G. Sirisha. Thank you very much for your meticulous efforts, relentless efforts for making this conference a great success. I should also register my thanks to all the moderators who have helped us to complete 101 scientific presentations in a span of 2 days. Simultaneously, 2 parallel scientific sessions were handled by the moderators. We should thank all the moderators. We extend our warm thanks to Dr. K.V. Sucharita Garu from SV University, Dr. R. Bharati Garu, Dr. M. Shiva Parvati, Dr. John Sushma Madam, Dr. Jay Madhuri Madam, Dr. C.H.M. Kumari Madam, Dr. R. Sujata, and Dr. Indira Priyadarshini for acting as moderators for successful completion of this scientific session. We cannot forget the people behind this. There are lots of technical and non-technical staff who cooperated for the successful completion of this. I want to make a special mention about Satyeshwari from Department of Home Science. She has helped us a lot with a lot of paperwork. Thank you. And we cannot forget the valedictory addressees, Professor A. Jyoti Madam and Professor Uma Maheshwari Madam for sharing your knowledge with us. Thank you once again for your presence and gracing the valedictory function. And I thank each and everyone for your patient cooperation, listening, and giving me this opportunity. Thank you. Hope we will meet in the next year's edition, NFFHH 2023. Am I right? Organizers should sell that.